So a very good morning to one and all present here. Once again, I welcome you all on the second day of this 2 days online workshop on bioinformatics and drug engineering, organized by Amity Institute of Biotechnology, Amity University, Rajasthan, in association with IEEE Rajasthan subsection. Yesterday, the inaugural ceremony of this workshop was started with a heartwarming welcome to all the dignitaries and participants by Dr. Manishita Das Mukherjee, Organizing Secretary, BDE, followed by address of Professor Dr. Vinay Sharma, Director AIB and Dean Research AUR, where he discussed about the human genome, bioinformatics, and also threw a light on the concept of drug discovery and various steps involved in it. After that, Dr. Rohit Bhakar, Chairperson IEEE Rajasthan subsection, outlined the role of bioinformatics in saving lives. He shared how IEEE focuses on the research and technology and also expressed his gratitude towards Amity University Rajasthan for collaborating with IEEE and also looking forward to many more collaborations in the near future. Professor Rakesh Bhatnagar, Honorable Vice Chancellor AUR, enlightened the audiences by sharing his past experiences before the internet era and how he was introduced to the field of bioinformatics that was also comprehensively explained by him. He also discussed the traditional methods of drug designing and how things have changed and have become more rational in the modern era. Dr. Manali Datta, Joint Secretary of the workshop GPE, expressed her gratitude and ended the inaugural session with the vote of thanks. First technical session of yesterday was taken up by Dr. Dinesh Gupta, Group Leader Translational Bioinformatics Group at the International Center for Genetic Engineering and Biotechnology, ICGEB, New Delhi. He also emphasized on the role of bioinformatics and artificial intelligence in COVID-19 research. Second technical session was a hands-on training session for all the participants where Dr. Puneet Singh helped all the participants exploring almost every feature of the Autodoc tool and how it can be useful for them in the future research. He also emphasized about the protein, the Chimera tool, and how we can use various features such as grid, ligand, etc. Dr. Ravi Ranjan, Joint Secretary DBE, also expressed his gratitude towards the speakers and the participants. So now I hope this second day would be even more fruitful and productive, and we would definitely be more acquainted with latest bioinformatics tools and techniques. So now let's begin with our first technical session of today. And I would take the pleasure of introducing our guest speaker, Dr. Ajit Kumar, Director, Center for Bioinformatics, Maharishi Dayanand University, Rohtak. Dr. Ajit Kumar is presently working as Director, Center for Bioinformatics, MDU, Rohtak, and has also been the Coordinator, DPT Bioinformatics Infrastructure Facility at MDU Rohtak, apart from being Deputy Director, Digital Learning Center at MDU Rohtak. He has about 20 years of teaching experience and has guided 5 PhDs and over 60 MSc, MTech, and M.Pharma dissertations till date. He obtained his MSc in Biotechnology from Punjabi University, Patiala, Advanced PG Diploma in Bioinformatics from Pondicherry University, and was awarded PhD from the University of Rajasthan, Jaipur. He has taught MSc students at HNB Garhwal Central University, Uttaranchal, Modi Institute of Technology and Sciences, Sikar, Rajasthan, and ITM Universe, Gwalior, before joining his present organization, that is Maharishi Dayanand University, Rohtak. His research interest includes toxicology, drug designing, QSAR modeling, virtual high throughput screening, drug repositioning, system biology, computational biology, pesticide designing, and biological database design. Presently, he has started venturing into big data analytics and cloud computing for text generation biological data. and is also working on different research projects funded by government agencies like UGC, DBT, etc. He has over 35 publications to his credits in journals of international repute and is also fellow or member of scientific organizations like Society for Science and Nature, Association of Microbiologists of India, International Association of Computer Science, and Information Technology, Singapore. He's also an editorial board member and reviewer for many international journals. So now I would hand over the session to Dr. Ajit Kumar. And I would like to tell all our participants that Sir is not well, but still he has agreed to join this session. So we are highly grateful to you, Sir. So now I hand over the session to you.
Thank you, Dr. Ravneet, and I hope I'm audible.
Yes, sir, you're clearly audible.
Yeah. At the onset, I'll say thank you to Dr. Sharma, Vinay Sharma. He is known to me since he was as a coordinator of BTIS network. And thank you to Dr. Manishita for inviting me for this lecture. And my own, I'll say, Dr. Ravi Ranjan, who has been associated with me since his PG, MSc days. And he did his PhD also under me. And Dr. Ravneet, thank you for introducing me. And nonetheless, I'll first in advance only thank all the team who is organizing this workshop. In this pandemic era, this way to connect to students and keep them updated is not only, it's something like we are taking a chance in the deprivation. Normally when we are in offline classes going on throughout, I had an experience that our students are almost interacting with 4, 5, maximum 7, 8 faculty members who are on daily basis. But this online mode has given a chance for all of us to make us introduced to almost all throughout the globe. We can connect to anyone, and we keep on interacting. Our students keep on interacting every time. So this is good, and I feel all the participants here should be very straight in knowing the things. Wherever, I hope you have collected a lot in last few deliberations yesterday, and since today is the first day, I'll see that a request to all the participants, mostly who are students and scholars, please be aware about and be wise enough to explore the things yourself also after this lecture or any of the workshop that you attend. So that will be giving you more of fruitfulness. So I will be starting the screen right now. Okay. And then I hope my screen is visible now.
Yes, sir.
Yes, sir.
Fine, thank you. So I wanted to discuss particularly because it is a workshop on bioinformatics and drug engineering. So that is one of the area where I do explore a lot. So that's why I thought to start with what are the basic principles that we approach in computer-aided drug design, and with particular reference to I'll be going about very highly used buzzword, that is VHTS, virtual high throughput screening, how it is being used in drug discovery process. So with this, I'll just start with, I hope I should remove this because it will be interfering in the screen. Okay. So if I talk about the drug, because it is now not a scientific word that is being used. Every layman in this pandemic has learned the word drug, medicines, disease, virus, your CT value, RT-PCR, all these technical terms are on the common lips now. So as far as this drug is concerned, if I talk about the life cycle, it is simple like even the normal any product in the market that is launched. Similarly, this drug is also a product for me, at least you can say, and even those who are producers, even the scientists, they also work with this agenda that drug should be taken as a product, and since every product has got its own life cycle, similarly drug also has got a life cycle. So I'll take you to a journey to this life cycle of the drug. And to start with, I'll say that it is almost 15 years behind the birth of a drug, and that involves screening and evaluation of some more than 10,000 compounds, and that involves somewhere above $800 million US dollars, that gives rise to a drug, that gives to a drug. And if I talk about the whole process, how it comes from the lab to the market, it takes if I go specifically in this, what is the reason behind developing a drug? So first, I will say a new disease that is very recent for us, we can say, like a new disease coming into the population. So like COVID-19 is one of the new virus that has come up, SARS-CoV-2. So that is a new virus, and the new disease COVID has come into market since 2019. So this has led all the scientists and the pharma companies also to work towards developing a new drug for antiviral for COVID as such. The second reason may be that there may be a drug failure in the market, which has already been launched and approved and launched in the market, but it failed at some time. Like remdesivir was one of the drugs that failed. It was launched with some another purpose and it failed. Now we see again, because of the another term that we have, drug repurposing or drug repositioning, that remdesivir is now being used as antiviral for COVID. So again, so this is one of it, which we talk about. If a drug fails, so we have to design another drug. We have to take it into consideration for that specific disease. Many times we develop some resistance. I will say the best example is your the third generation antibiotic that we had, amoxicillin, and we have a lot of beta-lactam other antibiotics for which we the bacteria developed resistance due to one specific gene that they had, beta-lactamase, that was evolved in them in the bacterial system because of evolution. So because of beta-lactamase, they used to cut the beta-lactam ring of the beta-lactam antibiotics, and these antibiotics were no more effective. So because of that, the resistance was developed, and then came the scientists started pouring in their head, and they found out the resistance was due to this gene, and they started targeting these genes, and that's there came your clovenic acid. So amoxiclave that we have now, the combination. So clovenic acid goes in, clovanate goes and bind to the target promoter site of these beta-lactamase gene, which doesn't allow the beta-lactamase enzyme to be produced, which in result makes our beta-lactam antibiotic effective against. So that is how we are crossing these resistance. So the third reason was about, I can say, is the resistance being developed to the existing drug that is there in the market. And obviously, this is the new thing that we say, new competitive drug that comes into market that leads other pharma companies to work on it. So if I have got a drug in the market in one of the company, so another company to counter that drug market, to occupy those share of the market, they'll start going up with the new development of a bit novel drug against the same competitive drug. So these are the basic 4 reasons which I find, because of which we have this drug discovery being started, launched as such, the process. And the process, if you see the drug discovery process that starts with NCE, that I say new chemical entities. So these are those compounds which we start working on. So new chemical entities, we have to have a list of new chemical entities, and we start finding out one of the molecule or few molecules which can be actually working as drug in later on stage. So the whole process where new chemical entities that we have designed or we have found is coming and is being converted to some of the drug-like compound is called lead molecule, and the whole process is called discovering a drug or drug discovery. Then from there, this process of drug discovery as such, if you see, is going to take 6 to 7 years. It was the time span being taken earlier. And then comes the phase of second phase, that is your drug developmental stage 1, that is also known as your preclinical stage 1. I say this is called preclinical because this is the stage when we evaluate the pharmacokinetic and pharmacodynamic pharmacological properties and the efficacy and the toxicity and everything on non-human subjects. Like it may be any model, mouse model or rabbit model or monkey model. So any primate model can be used. So these are preclinical stages where we develop the drug and evaluate the drug. And if they're effective there, then we term it as a prodrug. So stage 1, preclinical stage, talks about conversion of lead molecule term to a prodrug term. That means it will be working as a drug now. So, and then comes the last stage, that is your stage 2 of drug development, or so we say clinical stage, where we go and study further the pharmaceutical and pharmacological properties of that compound on primates. And then we shift to clinical trials. And again, I will say clinical trials is no more a buzzword now. It's a common word, phase 1, phase 2, phase 3, phase 4. And today, if we talk about government of India has lifted its certification on the vaccine, your Covaxin as well as Covishield. They had approved it as for emergency use. Now it has been it can be used without the term emergency use. So it has passed out the phase 4 also. So so this is what we talk about clinical trials. The trials are where we use human subjects and based on the population size and the target and the data that we are trying to evaluate. It is divided into 4 phases: phase 1, phase 2, phase 3, and 4. And if I talk about this development, design and discovery was okay. It took 6 to 7 years. The development process used to take is still taking 8 to 9 years of the time. So if I comprehensively talk about after this phase 4, they pass out, drug is launched into the market and is available for the patient. So the whole life cycle, if you talk, is about 15 to 16 years. Now the question comes, it is being done since the discovery of first antibiotic. People have been discovering and designing and developing drugs, and pharma companies are in the market and they are doing their business, they are manufacturing compounds. So what is the fun of doing or involving bioinformatics, particularly computer-aided drug design, into drug discovery and development process? So this is one of the very important questions that we should be answerable, and we answer it all time before we start learning it, why it is important. So here comes the timing, which I was talking about, 6 to 7 years. 6 to 7 years means here, if I can save using my band for tools or computer-aided drug design tools, if I save 1 or 2 or 3 years of timing here in drug discovery, where I discover a lead molecule. So before designing a lead molecule, if I find out, if I reduce my timing to say 3 to 4 years here only, I'll be launching the whole process 3 years before into the market, my product. So that is the advantage of CADD. Otherwise, there is no other differences. And why it is going to give you the help, I'll give you what is a significance, commercial significance of CADD is clear with this chart. If I see here, the same thing I've talked about. If you look into this side, it's the process of drug discovery is a high risk process. We start with a lot of compounds, say 10,000, 20,000 compounds, and we go for evaluation. We hardly a few compound reaches a clinical trial, and after that a few compound gets 1 or 2, or say they get approval from FDA in a year. And if you see, hardly of them 5, 6, or say 1/3, 30% of them are going to make money from the market. That is important, because any research involves money, and we have to see from where money will come, and we have to justify those money also. So that is more important as far as scientific arena is concerned. We have to work very specific there, and to make money for our organization, rather the commercialization of those compounds. So that is the importance we normally talk about. If I give you this, let me see if my screen will be clear. Okay, fine. I hope you have heard this word omeprazole. This is the compound that comes in the market with the trade name of OSID, Omaze. There are many other trade names. So this is one of the GI tract relaxant drug, and used for flatulence gas, peptic gas, and even your ulcerization and acidity, basically. So this was a drug being designed, and I term it as a blockbuster drug because if you look into here, the bottom line, I'm talking about, this is the data. This was introduced in the year 2000, and the data comes in 2001. This is the collection of omeprazole in 1 year, the year of launch. So in 2001, 2001 financial year, they collected somewhere $6.26 billion revenue, one drug, omeprazole, throughout the global collection. And if you compare it with the that time Hollywood hit movies that involves your Jurassic Park, Titanic, and all, so Star Wars, all those Bollywood, Hollywood hit movies, their collection was hardly 5.2. Global collection was 5.2. So it crossed somewhere above $1 billion collection compared to the market of Hollywood. Total market of Hollywood, if I talk about. So one drug, if it is going above the collection, so that's why we term it as a blockbuster drug. It is one of the reference compound that we take, and reference drug that was that collected money in 1 year beyond the collection of the total hit movies at that time. So that is the commercial importance. So if I'm saying this is a collection of 1 year, just imagine if I save my company, if I'm working for my company, and if I launch the drug, say 3-4 years earlier into the market, so I'll be collecting somewhere more than $25 billion extra, and that too, I'll be not spending much on. I'm reducing the cost of designing it and developing it also for 4 years. So these two collected together gives the importance of the significance of computational tools in drug discovery process. So if I what was the traditional approach? If I go back again, how did we started with drug designing process and development process? We remember the surprise discovery of penicillin, and so that was one of the traditional approach when we used to design and develop drug. like isolating an active ingredient from any natural compound, say penicillin or quinine coming from bark of cinchona tree and used as anti-malarial drug. Then we have we also used to have screening, broad screening for the anti-pharmaceutical activity of any compound, natural compound. Like we had a lot of sulfur drugs being synthesized and then evaluated for those activity, pharmaceutical activity, if I talk. Even we can go for modifying the existing compound, like we kept on modifying your beta lactam ring, and that converted from first generation antibiotic to second and third and fourth. So we have got different generation of antibiotic coming up. So we keep on modifying existing compounds which are already there in the market. And else, this is one of this another antibiotic, cephalosporin, that used the synthetic compound and used structure activity relationship basically to design a drug. Whatever the process be, if I focus here, these four processes, whatever these four processes will be approach for designing a drug or developing a drug, it is going to, if you see, we need a lot of time, money involved, because research input is required involved, and we need a highly trained manpower. So these things where a lot of input is required, and I've shown you how a result drug can be a blockbuster also. So because of these things combined effect, that came into that launched a battle between pharma companies, and they try to now rationalize their drug design process so that we can give lesser of input and maximum of output. So that was the concept of rationalizing the drug design process. So there came the first concept of rational approach of drug design, that is your modern days drug discovery process. So that talks about that basic principle, which we say is that was introduced and in structure with the concept of SAR, structure activity relationship. And I hope my screen is visible and audible.
Yes, sir, it is visible, but for a moment it was freezed, but now it is
Okay.
visible and audible.
Thank you. I thought, because I got information that my net is fine. So so that resulted into this is what we talk about, rationalizing the process of structure, the drug design process. And nowadays we talk in the terms of structure based drug design, instead of the concept of ligand based drug design is almost negligible now. So we don't start with the bulk of compounds and evaluate them. We know the target first, and if we know the target, by identify for that we should have a lot of scientific research incorporated in it. So obviously I'll just put it in quoted my own quote, that it doesn't mean that the earlier approach or the traditional approach of drug design were irrational. So they were also rational, but now we are more rationalizing it to make the process more efficient and commercially viable, but better to say. And if we go through the flowchart, how our modern drug discovery goes as such, we should, what is the basic requirement is now knowing the disease. So we start knowing the disease related genomics by using gene expression profiling, proteomics, genomics studies. And then comes to identify those proteins out of those genes and the expressions. I mean, when they're expressed, they will be giving us certain proteins. If not, we will have some transcriptomes. So either transcriptomics or proteomics we try to identify which can be our target for attack, so that we can counter those targets. Like we have spike proteins in COVID, your SARS-CoV-2. They are the site of attachment. So if we target those ACE protein or the spike proteins of virus, so these two become our target proteins. So that way we identify those targets by using, again, bioinformatic tools, reverse docking, protein structure predictions. All these things are of info tools. So that is required here to identify the target. Then we need to validate it. So validation, the target which we, out of all the proteins that we have deciphered involved in a disease, and identified one or two of the target proteins, we need to validate whether these proteins are really the target by using testing them for druggability. Are they druggable in nature or not? Whether they can be targeted with a drug or not, whether they can attach to any drug or not. So one thing that another is your inhibition assays or gene knockout or knock-in technology we can use. So these are wet lab again coming into play. Then again, it comes down to dry lab, where or bioinformatics, where we go for discovery of the lead by using library design. We modify our compounds. We design our compounds based on pharmacophoric patterns deciphered from the receptor. And then with this, we either screen out and take some hits from the databases that we already have, chemical databases, which work as a library, or we design in ourselves also in lab, that is using combinatorial library approach. So we prepare a library of chemicals. We use them against our target, validated target. So using for that, today we'll be discussing that, how to do it using molecular docking. That is one of the tools that we use for VHTS, virtual high throughput screening. And then we can have pharmacophore studies also, and we can modify certain compounds with certain side group modification, which can again further lead to the enhancement of the activity of that compound. So these things, apart from that, just hold on. Yeah. We have that is the next stage, where we optimize our lead molecule to enhance the pharmacological or pharmaceutical property of that compound by using your QSAR methods. Quantitative structure activity is another tool, quantitative structure activity relationship, which is, we say, structure based optimization. And then we also modify them according to their pharmacological properties, like your ADME, absorption, distribution, metabolism, and excretion, apart from toxicity. So we profile these pharmacological or toxicological profiling, and then in silico. And then we try to modify, optimize those side chains, if the side groups, if they are hindering it, and even elevate the property of the compounds, pharmaceutical property of the compound, so that these leads can be treated as a prodrug. And that leads to the preclinical trials, where we go for pharmacokinetics and pharmacodynamic properties in analysis, admit profiling using animal models. Then that leads to the final stage, that is your clinical trial stage, that is your human stage. So that already I've discussed in your life cycle part. Then comes, so this is the same graphical vision of diagrammatic vision of the process which I've talked about. Here we go for the molecular studies, and we design the clinical features which will lead us to investigate into the molecular level studies for identifying the cause of the disease. And using them, we try to find out the target site or the target protein, where we can use animal studies for that. Can be of transgenic knock-in, knockout, any mouse, or conditional knockout mice model. Even we can have antibody, antisense, or interfering RNA technology being used for those things. So using these things, we identify our target. And then when we have got the target, we go for these ligand binding. And this target normally are receptors, your ion channels. Basically, they are membrane bound proteins. So there comes another role of in silico, where in vivo or in vitro job is not, I can't say it is, it cannot be done, but it is very tedious. Because membrane bound proteins are very difficult to be crystallized and analyzed as such, and to be, or rather to be isolated in intact form and studied. So that is where the role of molecular modeling comes into play, because of maximum of the targets are your membrane bound proteins, and these membrane bound proteins structures are not so easily available, and we cannot work so easily on these proteins in isolation. So that's why your in silico techniques come into play, and where we can easily handle those things in silico using molecular modeling, homology modeling, your de novo modeling, and if any, as per the requirement. And that will help. With this, we have another list of techniques which will lead to identify our own lead against those targets, identified and validated targets. Which one is the lead compound? And then comes the optimization of these lead by selectively changing or altering certain of the side chains. So this whole process looks so easy to go, but it is a bit of iteration. Many times it fails here also, and again goes back. It keeps on iterating a cycle before it reaches your as the candidate becomes a pro-drug candidate and is evaluated for safety on the mice model or the mammal model or the primate model, and then further going for human studies or clinical phase and getting an approval. So with this, I will skip these slides because I already have discussed about. Yes, I've talked about these testing. I'll come down to one of the topic which is so important as such. VHTS. I hope you have yesterday also gone. I was talking to your organizers that you people have got a feel of what is molecular docking as such. So this is one of the technique that we use in VHTS, virtual high throughput screening. High throughput screening, HTS, is the part of wet lab where we use robotics, and we evaluate say 1,000 compounds in one go on a cell line. But at the same time, when we evaluate the same 10,000, 1 lakh compounds in silico on computers, that is termed as virtual high throughput screening. So that gives an edge to us to evaluate them at more of molecular basis. So as the term says, that it is an established methodology to identify a drug candidate from a large collection of compound or a library. And it complements the expensive and time-consuming process of HTS. So although it has got certain of the drawback, not I can say the drawbacks, the challenges are that it is again predictive in nature. So prediction always remains as such. Just wait. Yeah, hello. Morning. Yeah. So, and this, if I talk about that, the challenges which is being posed by docking as such or VHTS, the successful, how do we overcome these challenges can be by using, by taking care of the whole process of docking VHTS, high throughput screening at every stage, right from the start, identifying the compound. When we identify the compound, we see the compound and we design it, or rather we get the structure of it from the drug structure data bank. and we analyze those, their poses, their binding site, on that protein, on that receptor. These things are important right from the start. If we are modeling them, if we are modeling them, are we getting the correct model or not? That is important. Okay, so at the same time, so that is one of the important feature. I mean to say the important parameter that has to be taken care of right from the start when we are designing the ligands also, because these ligands are supposed to be designed or in your library, the chemical library that you take. So these design process of chemicals, the optimization process, the energy optimization process, the algorithms that we run for optimizing the energy of these ligands, the algorithm that we have to use for optimizing the energy of the protein as such, these things are supposed to be very importantly taken care of. We have to take care of these things very right from the start before we go for the docking algorithm. And the docking algorithm also, we have to see which pose we have to go for, how they are going and attaching. Analysis of those docking pose are very important critical, because a molecule can be inactive if it is a bit of different bioenergy level. So that is not becomes your bioconformer. So when we are into these process, we cannot just use as such, because if I compare it with simple word, window word that we use, if you type something on your keyboard, you're giving an input. So you just start hitting your keyboard, it will be showing certain output on the screen. So whatever be the input, there will be certain output because it is going through an algorithm process. Same thing, it happens with docking also. Whatever you give as the input, they will give you certain output as the output of the algorithm. But are you giving the correct input so that you get a correct sentence or the word being framed? So the sequence of input and the sequence of output are strictly correlated. So that is where we have to take care of. So that is what I say here. The successful VHTS requires a careful implementation of each phase of computational screening experiment, right from the target preparation to the hit identification and lead optimization. So this funnel that we talk about is very important to filter out our molecule, which we really get, and we do it very efficiently, and we have to. So now going to the next one, if I talk about what is molecular docking, it is a as such fundamental tool of virtual high throughput screening, and you see as the picture is, it is something like a puzzle game when we try to attach. So it is basically, I talk about it as a problem. It is a problem, and we have to see, we give child to go and click which pose is best to attach. So that way a child develops it's a brain game that we give to a developing child. So same thing, how it is going to match. So docking is basically a structural technique which is going to find out which way your small molecule is going to attach and find the correct pose into the receptor, that is your protein molecule. And this orientation of attachment is going to give the stability of the complex. And more is the binding, better will be the activity, biological activity. And biological activity here, if I talk about, is nothing but your affinity of the molecule towards the target molecule, a target protein. So this is being used as the scoring function, how they are posed, how they are attached to it each other. And this is what we said, target binding affinity or binding score or docking score, that we other way technically say. So these are the few terms already you knowing, but again, I'm revising it, like that we always use in glossary of molecular docking, that it can be the receiving molecule, mainly it may be protein, even your DNA many times nowadays, even RNA. So nucleic acid or protein can be your receptor, host, or the lock. And the ligand that you attach small molecule that you want to see the binding of is being talked about as ligand, guest, or the key. And docking is a process of computational simulation of a candidate ligand binding to a receptor, while the binding mode is the orientation of the ligand relative to the receptor, as well as the confirmation of the ligand and the receptor when bound to each other. So in which pose, in which binding orientation they're binding, I mean to say the ligand and the receptors, they're binding to each other. So on individual binding orientation is termed as one pose. And the energy that is being released during the binding is calculated as a binding energy. And that talks about more is the attachment of the molecule to molecules, more will be the affinity, and greater will be the energy released, and that will be used as lower will be the binding energy, or the binding higher will be the binding score. So this is what the scoring function talks about, and based on the score we talk about the ranking also. So this is the two types of docking that we normally use. Nowadays we have got protein docking also coming into play. So otherwise we normally have, when you are using your drug discovery process, it is basically molecular protein and small molecular docking, a small molecule docking that we use. So if I define as such the problem of molecular docking, I say it is a problem because it is something like a puzzle and we have to solve that problem. That puzzle has to be solved. So there are two types of approach that we look into. Similar that we have studied in your enzyme kinetics or in the mechanism in the enzyme action. It can be either lock and key or induced fit. So we have lock and key docking method also, and that is your rigid and rigid docking. And second type of docking is your flexible docking, where we can have a bit of flexibility in the protein and higher flexibility in the ligand. That is called hands in glove or induced fit docking, binding docking of the molecule. So these two are different types of docking methodology or algorithm that we have, rigid and flexible. And this is what we say shape complementarity approach. This is one of uses the matching technique that describes the protein and the ligand as the complementary surface straight, and we try to calculate the this complementarity of each portion of the receptor and the ligand, and try to fit into where they are going to fit. And this specific orientation is defined and collected as one of the pose, and that is taken as one of the, and the energies are calculated, which will give you as the energy score. As we can even have the simulation covering up, where we actually simulate, that is called ensemble approach, where we simulate the actual docking process in which your ligand and the protein, they keep on interacting one individually in a specific zone of the binding area, and the energies or interaction energies are being calculated. Okay, so and these, what we do here is basically we are trying to calculate the hydrophilic and hydrophobic interactions, the number of hydrogen bonds, whether they are accepted or donated, and what are the orientation of the ligand, and with these what all are the other forces, non-binding forces or non-binding forces, non-covalent binding forces or covalent binding forces. We calculate all those things to finally come up to the release of free energy concept. So how much free energy is being released out in going for that binding, and that calculates your final dock score or the binding affinity. So this is all I have discussed about, nothing to say as such. I'll give you a bit of, there are few softwares which are free in and available for you, which uses molecular docking algorithms. Sanjeevni, it is at IIT Delhi developed, and it is a good, very good software. Gold, we have Autodoc, normally people use. Then we have got GemDoc, Hex, another protein docking. For protein docking, Hex is normally used, and then Gramex we also use for protein docking. So with this, I hope I have come up to the end of my talk, and I'll certainly ask you to if any doubt.
So there are a few questions which have been posted in the chat box.
Okay.
So sir, we'll take them one by one. So first question is by Miss Aditi Nag, and she is saying, sir, with modern drug discovery approach, how fast can we expect a drug based on quinic acid discovered by IIT Mandi and ICGB against SARS-CoV-2?
See, as such, if I talk about the drug discovery process, the government is doing very fast in approving the process of approval and looking into the pandemic scenario. But as far as now, I see the graph of pandemics is also going down. The regulation will also be strictened up. Earlier, it was given very easily. So even the vaccination that we have, if you remember, particularly, I'm not answering this. I'll give you an example too. We have that vaccine coming up from Oxford, and that AstraZeneca that we have all been injected with, Covishield. So that that was developed already in the month of March 2020. So it was into clinical trial phase 1. So if you remember the quickness of, so they had developed it for SARS virus, but now and with SARS-CoV-2 coming up, coronavirus variant 2 coming up, they just had nothing doing. They didn't add anything. They didn't delete anything. They went it directly and they tested it for CoV-2 also, and they found it okay. So it was introduced and it was into clinical phase 1, phase 2, phase 3, approved. Phase 3 was approved, and government gave them the approval for emergency uses. So still we all have been vaccinated under the category of emergency use. So it's not still approved by the government agencies. So, and as far as the drug is concerned, there is one antiviral drug. Antiviral drugs are not so easy to develop, basically. That is because these viruses keep on changing their target protein so quickly that even now you see the virus CoV-2, COVID as such, has changed a lot, and it is still changing. And the beauty of the virus is that they want to cohabitate the globe. हमारे साथ वो भी रहना चाहते हैं, वो हमें मारना नहीं चाहते हैं. So that's why the lethality of the virus strain is going down, and we are sharing now. The virus is also sharing the globe Earth with us, and they need us as a harbor. If we are dying, if we will be dead, they won't get a place to survive. So the virus will also be dead. So everyone wants to survive. Even the viruses wants to survive. So they change themselves. And if we want to attack them, just to survive, they are changing themselves. Just think of if we are trying to attack them, they will change so quickly that they won't be available as a. We have to keep on checking the target site. But although there are certain comprehensive drugs, but these drugs are having a lot of side effects, antivirals. So that is one of the hitch where we have to overcome as far as the drug discovery and designing process for viruses specifically is concerned. We can have vaccinations so that our own immune system can respond. So vaccination is successful for antiviral diseases, viral diseases. But treatment as such, antiviral drugs are not so popular and not so effective. They are supposed to be used only in the life and death situation. So yes, we do have drugs, but we should not rely on antiviral drugs so much.
Thank you, sir. So the next question is by Abhishek Chahar. And he,
Dr. Manishita,
yes,
sorry
ma'am.
for the interruption.
Yes, ma'am.
Before taking up the next question, since our Honorable Vinay Sharma Sir, Director and Dean Research, he has joined us. So firstly, I would like to invite Sir for his kind words in this technical session.
Yeah, thank you. Thank you, Dr. Ajit, for
Thank
a
you.
very comprehensive talk.
Thank
I
you, sir.
joined late, so at the outset, I would like to extend very cordial welcome to you.
Thank you.
That you took time out to give a talk to our audience on this 2 days workshop.
Sure.
It's also very good to see you after a long time, I think several years.
Yes.
Because of COVID, and then, the suitable platforms were also not available.
Yes, sir.
So it's very nice to see you again here.
Thank you, sir.
And I find that the talk is very apt on a very appropriate title related to the virtual high throughput screening, because that is where the bioinformatics is aiding a lot, because earlier we had this chemical screening that, a lot of molecules had to be synthesized and then they were to be screened.
Yes.
So naturally the cost was highly prohibitive.
Mhm.
And it was time consuming also. It requires a lot of manpower as well. And I think that is where the bioinformatics has made the life easy for us. And we are able to now, through the virtual high throughput screening, we are able to find a few molecules which can be considered as the candidate molecules or the lead molecules. And then we can go in for the experimental validation. So you have given a very comprehensive talk also on the related aspects. So I feel that it's very good for our fundamental, for our basic learners as well as the advanced learners. It is useful to both the categories of participants. You've built up a story starting from the beginning and then, moving forward and talking about the nitty-gritties of the bioinformatics applications in this very important area. So I think the session can continue, and over to Dr. Ravneet. So you can continue with the session. I think a lot of questions have come up because it has been a very exciting session,
Yes, sir.
thought-provoking session. I was looking into the chat box and there are quite a few questions. So I think let's go ahead with the session. I think that's more important.
Yes, sir.
Because
Thank
we
you.
can talk on and on,
Yes, sir.
Yeah.
Thank you so much, sir. Thanks for coming.
Thank you for your time, sir.
Thanks, sir.
The next question is by Abhishek Chahar. So he's asking, so how can we use NGS other than identifying target sites with respect of drug discovery?
Yeah. NGS, as far as the target site is concerned, it is more of importance in identifying the variations in the genome of, like, if you talk about in viruses, in specifically I'm talking about COVID, how they're shifting, how they're changing their genome. Their NGS is important. Else, other way around, if I talk about, it is more specific for, we say, pharmacogenomic studies. So where we give gene therapy specifically. So like NGS helps us. I will give you one small example. Even the antibiotics or even any other drug we use has got different efficacy across the globe, depending on the human race. So because there comes the role of NGS, and if we have got our own gene, the target gene being specified, it may happen that cephalosporin type of antibiotic is active in North India. At the same time, beta-lactam antibiotic is effective for the same disease in South India. So that is not only the way we talk about, it's the two-way process, the host as well as the causative organism. both way we have to study, and obviously NGS is going to help to identify the target sites and the variations in the target sites. That's important. I hope I have answered.
Yes, sir.
Yes, sir. Thank you, sir. Sir, the next question is by Miss Ruchi Kumari. And first of all, she has thanked you for the wonderful lecture. And then her question is, I want to know that if I want to design a drug for a cancer treatment, then what should be my first step towards it? Like, I am very much interested in learning drug designing. So how should I proceed in this field?
See, cancer is not a one process disease, and which type of cancer you want to work on first thing, because we have got different cancerous genes being expressed in different part of the tissue, body tissue, and depending on that, we have got different types of drugs, cancer being developed. So, which cancer you want to work on? Is it a comprehensive one? Second thing is that the cancer specifically, if you have to work on, I have to start with the learning that the molecular biology and the target sites of those cancerous people have been working in cancer biology. And the biological cancer biology will give you the list of proteins and the target sites to, and not only proteins, we have got the genomic sites also where to target them. So these targets are being identified through in silico techniques. So that is where we can help. You can take the help of, and if you're really working on interested in, you can use phytochemicals analysis for anti-cancerous activity. That will be more important. I'll suggest you that way. So
thank you, sir. The next we have the question from Krunal Pawar, and he's saying, how can we use epigenetic biomarker like non-coding RNA or circular RNA as targets?
epigenetic biomarkers are not always the biomarkers in every case, because epigenetic diseases or the targets, these biomarkers can be only referred to when we don't get answers from genomics. So it is not the answer always. There are a lot of disease where we have got epigenetic biomarker, epigenetic cause, and where we can develop epigenetic biomarkers. And even your non-coding, what you say, non-coding RNAs, even and your circular DNA RNAs, they can be also working as the target sites. You can, as you said yourself, biomarkers can be used as the diagnostics, not as a treatment profile, which I feel so.
So next we have another question from Deepshikha Gupta. She is saying, what should be an approach to check for protein targets of newly synthesized heterocyclic ligands to have drug-likeness? How can we search PDB files for checking interaction studies? Yeah.
Okay. So she's very specific, and the answer will be very specific. See, if you have newly synthesized, you have got heterocyclic ligands compounds library. The two-way approach is, first you can go for 3D QSAR COMFA studies, QSAR studies. Another is your virtual screening method that we have discussed, the molecular docking studies, by identifying their targets. And before that, you want to know whether they have got drug likeness. For that, I'll say you should go for, there are Lipinski rules first, to evaluate them for Lipinski rules. Second, then you can go for their softwares admit SAR properties. So using admit properties, very soon we are launching one of our very good web tool for evaluating the blood-brain permeability specifically, and they are going to also calculate ADME property. And I hope we are waiting for, we are into communication with the reports. I've already reported it. So the day we are published, we'll be launching that site also. And I'm very happy that we have got a high percentage of positivity report as compared to the earlier only one that we had was admit SAR. ADME SAR website, or the tool that was used for calculating the drug likeness and toxicity profiling. And so we have developed it based on machine learning tool. So it will be launched very soon. So that also you can use next time, or else if you want, I can give you the straight, you can connect to any of your teachers there. I'll give you the link. You can evaluate your molecule directly from our server right now for the time being, before it is getting public. And if you want to check for the, you have to first see the protein targets. Are they available on PDB or CSV? If their structures are available, download it, optimize it, and then go for energy optimization first, and minimize it, and then go for binding, molecular docking using autodock or hex or anything. So that can be done.
Sir, there are still many more questions, but we are running really short of time.
Any important, let me check it out.
Yes, sir, we can just take one question of your choice.
What is you considered a good range of binding energy or stable interaction? It should be somewhere in the range of -5 to -9. I feel so. And natural compounds, most important. And then that too also we have to look for the runs. How many runs we have gone for by docking. So if it is say 1,000 runs, sorry, 100 runs. So we should get at least a bigger cluster of 20 to 25 for that, to use that as a binding energy rate. And then natural compound based on drugs. Yes, you're correct. Natural compound based drugs and synthetic drugs undergo the similar process of drug discovery. The only difference is that we need to know the chemical composition of that natural compound. Okay, fine. I think epigenetic. I think I have answered.
Thank you, sir. Almost you considered all of the queries from participant. And one thing I can promise to participant, that if some question is left unanswered, that we will take it maybe through email or something that. And as soon as I receive these queries, I may ask you to please answer this on email. So because of time consent, and I understand that you also have some other engagement in your university, so we have to proceed now for next session. So I think Dr. Manisita, it's the right thing that we can do for these queries.
Yes, sir.
That is
Yes,
unanswered, right?
Sir, on behalf of the organizing committee of BDE 2022, I extend my heartfelt thanks to you for sparing your time for us. And sir, I myself am recovering, so I really know the condition in which you are in right now. And sir, the way you delivered your lecture, it is evident from the questions that how interesting your lecture has been. So, sir, thank you so much. Like, a thank you is a very small word to appreciate the work you have done, sir. Thank you is all we can say. And hope to continue with this interaction in our future also, sir.
Thank you.
And we love to have more lectures from you. Thank you so much, sir.
Thank you, Dr. Das. See, I will say one, I'll just add to which Professor Vinay will also agree to. We all are teachers and we keep on deliberating lectures even on our deathbed. We can do so.
You're absolutely right, sir. Thank you so much.
Thank you.
So, Dr. Manishita, can we move into the next session?
Shall I leave? Can I leave if it is so?
Yes,
Yes,
thank
sir.
you, sir.
Yes, sir. Thank you so much.
Thank you all. Thank you.
Participants, we are continuing with our next session, and Dr. Poonam Vishwakarma is already here. So, Ravneet ma'am, I hereby request you to please take over the session. Thank you.
Yeah, sure, ma'am. Sure.
Dr. Poonam, can you hear us?
Yeah.
Yeah, so welcome to this session. Dr. Ravneet, you may proceed with further session.
Sure, sir, sure. So, welcome, ma'am.
Thank you.
So, after this informative talk by Dr. Ajit Kumar, once again, I, Dr. Ravneet, welcome all the participants in the second technical session of the day which will be a hands-on training session mentored by Dr. Poonam Vishwakarma, postdoctoral fellow, University of Paris, France. So Poonam ma'am did her PhD from Jawaharlal Nehru University, New Delhi. During her PhD, she rigorously worked on membrane proteins. Currently, she is pursuing postdoctoral research as a bioinformatics engineer from the University of Paris. In her postdoctoral studies, she's working on the usage of nanobodies in the treatment of diseases. Along with the aforementioned, she's also involved in a COVID-19 project over there in France. She has published many research papers in journals of international repute. She has also developed a Docker-based web server by the popular name that is the ABC Finder. So with these few words, I would now hand over the session to Dr. Poonam Vishwakarma.
Thank you, ma'am, for such a nice introduction. Am I audible?
Yes, ma'am, you are audible.
Okay. Okay, I will share my screen.
Yes, sure.
So is my screen is visible now?
Okay, Dr. Poonam,
Yeah.
with your permission, I would like to speak to participant about this technical session. So this
Yeah.
technical session is of 1 hour, and we will resume this, hands-on training after, post-lunch session also. So due to the, time consent of speaker, one of our speaker, we divided this into 2 sections, and so this session will continue till 1, and then post lunch from 3 to 4 we will again resume this hands-on session. So I think in this session Dr. Poonam will discuss about homology modeling, right? Anything else
Yes.
you are discussing in this session, Dr. Poonam?
No, today I will, in this session, I will take the what is homology modeling and basic and hand-on session of the homology modeling, and evaluation of this predictive structure I will take to the next session.
Right. So session is handed to you.
Okay.
All the best.
Thank you.
Yeah.
Thank you. So today I'm going to discuss about homology modeling. So before coming to the main topic, I want to discuss why we are doing the homology modeling. What's the need of doing this homology modeling? So I will discuss the 2 points regarding this, why we are doing this. So there are certain reasons in the X-ray crystal structure where residue are missing, and we have to rebuild that particular portion. And if the residue missing number is 3 to 10 only, then we can rebuild that that portion using the certain software like PyMol. Am I audible? Okay.
Yes, you are audible, and now you are visible. For a moment, you were not visible, maybe camera,
Okay.
but
Okay.
now you are
Yes.
visible and audible.
So if in a structure there are only 3 to 10 residue are missing, so we can build those particular reason using a certain software like PyMol. But what happen when we have more than 10 residue missing? Like there are particular portion of the sequence is missing from the structure. That means that portion is not resolved using the X-ray crystallography structure. So in this regard, we have to go for the different approach. So in that case, we use the homology modeling when there is the certain portion of the sequence which is not resolved by the X-ray crystallography method. And the second thing, suppose you have the amino acid sequence. Here you can see the amino acid sequence. And for this, if you have the amino acid sequence, you have the structure of the protein. But if there is certain situation, and just a minute. There are certain situation case where you have the sequence, but you don't have the structure. So what we can do when we have only the sequence and no structure? So how we can determine structure in this case? So we can use the experimental method, which are the protein structure determination method. And these are the X-ray crystallography and NMR. And these are the time consuming as well as costly. So in this situation, what we can do? We can move for the computational based method, and using the computational based method, we can predict the protein 3-dimension structure. How we can do the computational based method? Suppose you have the sequence, we can do first the blast. Using the blast, here using this blast, we are just doing the multiple sequence alignment of the target sequence with the sequence which is already available on the particular database like the UniProt and SwissProt, and we are getting the high identical sequence similarity. Using this high sequence similarity, we are predicting the protein structure. Hello? Hello?
Yes.
Yes, ma'am.
Hello?
Yes, ma'am. Poonam ma'am, am I audible?
Am I audible?
Yes, Poonam ma'am, you're audible.
I'm not.
For a while, you were disconnected, but now you're audible.
Okay. So I'm not able to hear, am I audible or not? I think there was some network issue, so I'm not able to hear your voice. I don't know, am I audible or not?
Ma'am, you can
Okay,
continue with the session.
let me resume. There is some network issue here. Okay, so using the computational method, you are having the high sequence identity sequence. So what is the basic behind this? If two protein have the high sequence similarity, then they can have the similar 3D structure. So that is, if one protein like this template protein has the known structure, then the structure copied to the unknown protein with high degree of confidence. Here in the homology modeling using the computational method, we are just transferring the coordinate of known protein onto the unknown. These are the knowledge based method, that is the homology modeling and the threading. Homology modeling, in the homology modeling, we are determining the structural model based on the experimentally determined structure. And in the threading, we are determining model by structure similarity with or without detectable sequence similarity. So in homology modeling, we are using different types of tool like modeler and different types of web server also. Here today we are discussing about the Swiss model. The modeler is the standalone tool which we use for model the 3-dimensional structure of the protein. This is the Python based tool, and we need certain certain knowledge, small knowledge of the Python and the Linux based environment, but it also gave the very accurate result for the modeling the 3-dimension structure. But today is the online mode, so we will discuss about how we can build the protein 3-dimension structure using the Swiss model. So Swiss model is the fully automatic protein structure homology modeling server, and it is accessible via the XPC molecular biology web server. These are the certain steps. Further, we will perform all these steps. Here, I am giving just a brief introduction what we are doing in the Swiss model web server. So these are the steps like we have to input data and template search, template selection, model building, and model quality estimation. So the input data, there should be a certain format for the input data. That is, it should be in the FASTA format, cluster format, or Uniport accession number. You can put all these things as the input data. If you have the FASTA sequence, you just have to put the FASTA sequence, and there are certain steps, and it will give the result. And Uniprot ID accession, if you have this, you can put this. And second is the template search. What approach that this Swiss model use for the template search? So it's used the BLAST for the close homology and HH-plate for the remote homology. Third is the template selection. For the template selection, it used the QME score and QSE score and Blossom 62. So this is the approach for the template selection. You will get the different template, how you will select different template, what are the different approach. So this is the global model quality estimation score, and it reflect basically the expected accuracy of that alignment, and it's express the number between 0 and 1, where the higher number indicate the higher reliability. While we will do the workshop, we will again see what is this score and how we are selecting the template. Third is the model building. So it used the ProMod 3 engine for the model building and transferring the conserved atom coordinate. We can do the loop modeling for the insertion and deletion, and construct non-amino acid side chains. The fifth step here is the model quality estimation. So how we can choose our the model quality, whether it's good or not. So there is the Q-mean Z score, which represent the value of this Q-mean Z score is around, here we can see, this is just an example from 0. The 0 indicate the good agreement between the model structure and the experimental structure of the similar size. Model of low quality typical has the score of minus 4.0 or lower. So in this, hello, I can't hear you all. So I want to show how we can do this Swiss modeling, but I can't hear. So I want to show the demo. Hello.
Hello, Dr. Poonam. Hello, Dr. Poonam. Can you hear us?
Okay. So here I will show you how you can use the Swiss model to generate the 3D structure of the protein. So you can directly go to the link of the Swiss model. Here I just click on the link. So this is the page of Swiss model. And this is the protein structure homology modeling server. So here you have to click on the start modeling tab. Okay. Now here you just there are two options, like as I mentioned before in my slide. Yes, input data. So here you have to give the input of the data. So it should be in the FASTA format, it should be in the cluster format, or you can give the Uniport SSN number. So here I already downloaded the sequence of hemoglobin. So I will just I copied this sequence from the Uniprot. So you can download any sequence, FASTA sequence, for which you have to build the 3D structure of the protein from the Uniprot database. In the Uniprot database, like this is for the hemoglobin subunit alpha. So you have to go down, and here you can see the FASTA sequence format. You just have to click here and you can copy this sequence from here, and you have to paste the sequence here. Here you can just put the PDB Uniprot ID also. In the top, this is the Uniprot ID P09904 for the hemoglobin subunit alpha. You can either use FASTA sequence or the Uniprot ID. So here I just submitted the FASTA sequence. Now you have to click on the search for the template button. Once you have done this, you have to have some patience because it takes some time to generate your list of template. Once you have done this, you can align as many of this template to your target sequence. I think it will take some time, but I already done this, so yes. So as it is here it is running, you can see the template result while it is running. It is searching for the template. It is using the BLAST and HHBLIST again the SMTL. So it will take some time, and this is the result after it will give the search for the templates. So these are the list of template which switched model gave you. So here you can see the coverage, QM, QE score, identity, method. So these are the list of template. So this blue bar here represent the coverage. When you will click here, you can see the coverage that is here for this template is 1.00. That is it's covering the 100% of your sequence. And second is you can see the identity method. X-ray, the structure crystallographic method is X-ray method, and the you also along, so you have to make sure of taking all this point, not only the coverage, not only the identity, and not only the resolution. You have to select the structure with good resolution and with good coverage and with good percentage identity. So here you can see this is although this is showing 100% coverage, but the resolution of the structure is 2.2 angstrom. So you have to take the structure with a good resolution. There are different lists here. You can scroll down and see all this parameter, but the top are the good templates. Among the top 5, you can see the with the best resolution and identity and the coverage. So I had selected the one for this. So these are this is the template structure. So you can select one template structure, 2 or 3 as you like. So for this, you can see this is 100%, it's covering the 100% coverage, and the sequence identity is 85.11 and is X-ray crystallographic, and the resolution is 1.7 angstrom. While selecting the template, you should consider the resolution also along with identity and the coverage. Now you have to click on the build model. I think this also done. So you have to click here and you have to click on the build model. So it will also take some time and it will give result like this. Okay, we can wait for some minute. It will give the model result. Yes. So here is come. This is the model result for our query sequence. Here you can see this is the result and this is the structure. Here when you scroll down, you can see the alignment of the target structure, this target structure and your template, and the red color here which are showing less sequence identity, and you can see this red color is the N terminal or C terminal, and that's why it's showing the less similarity. Maybe that some residue are missing in the template, that's why. Otherwise it's the 100% coverage, it's showing 100% coverage. And the first thing after this result, you have to look for the QMQE score and QMIN disco score. So what is this score? This QMIN and QMIN disco give the overall model quality. You have to look first this two score, overall model quality, and its measurement between 0 and 1, with higher number indicating higher expected quality. And this QMQE is the coverage dependent. What is meaning of this coverage dependent? Like model covering only half of the target sequence. Like while we are doing this, uh, yes, you are the first score which we are looking here is the QE score. So the first graph, bar graph with blue color is showing the coverage. So this QMQE score is the coverage dependent. For this template, like is showing the 100% coverage dependent. But while you scroll down, you can see this is. For example, if it's cover only the 50%, then model covering only half of the target sequence is unlikely to get the good score, that is above 0.5. So this is score therefore along with this QMQE score, you have to consider that Q mean disco score also, because this is not dependent on only on the coverage. This is the other way you can evaluate your model. So is this Q mean disco score is the average per residue score, and which has been found to be correlated well with the IDDT score. And this provide error estimated based on the global estimation. And third is the okay. This is the Q mean Z score. I can't zoom it. But in Swiss model, you will get the Cumin Z score also, which represent the estimation of how comparable the model is to the experimentally derived structure. And this Cumin Z score around 0. Here you can see the score Cumin, that is minus 0.56 for this, and C beta is minus 0.83 for all atom solvation and torsion. So this Q mean score around 0 indicate the good agreement between the model structure and the experimental structure of similar size. And model of low quality typically have the score of minus 4.0 or lower. So if you are having your template selection and the Q mean score is around minus 4, so you can't take those as a template. So you have to consider this Q mean Z score also. If it is low quality, it will have the score like minus 4. And this is the normalized cumin Z score. Here you can see this red star. Here your model structure lie in this region. The Z score, this color represent the different Z score. So the Z score with greater than 2 is light gray. Z score which is less than 1 is less right, and this dark gray is less than 1. So if your model is lying between this, so you can consider this as your template structure. If this red color here showing your model, and if it is lying between this white region, so you can't consider those as a template. So you can consider these as your template structure. So while you will select, the template selection is the basic step for the homology modeling. So this is the main criteria, main parameter on which you have to focus while you using the Swiss model as a web server for building the protein 3-dimensional structure. This cumin Z score, QMEQ score, along with this score, cumin disco score also, because this is the coverage dependent and this is the coverage independent score. So after all this thing, you can, there is some option here, like you can download the PDB structure from here. And after this, you can download this model if you are okay, like yes, you further want to do the drug discovery with this model, docking and molecular dynamic simulation, whatever analysis you want to perform after this, you can download and do the further analysis. Before that, you have to do a structure assessment for this model using this template. So you just have to click here, structure assessment in the Swiss model page only. Okay, so it will give in the Swiss model, it will directly, you just have to click there, and it will directly link to the page of structure assessment. And you will see the Ramachandran plot and other property here in down the page for your structure assessment. So this is the Ramachandran plot. Ramachandran plot is a way to visualize the energetically favored region. In the Ramachandran plot, they are favored region for the backbone dihedral angle of the amino acid residue in the protein structure. This is the build structure of the unknown structure from the sequence using that template. So how you can check your structure, build structure is accurate or not. So these are the different structure assessment parameter you can also check. So once is the Ramachandran plot. So in this graph, they are white area. You can see they are white area. This is the phi and psi angle. They are white area, which this white area correspond to the disallowed, not disallowed region, where atom in the polypeptide come closer than the sum of the van der Waals radius, and this region are sterically disallowed region for all the amino acid except the glycine, which is unique in that of the lack of the side chain. And you can see that there are different color, like dark green, light green, and they are lighter than this green. So the dark green area region conform to the allowed region. Here basically the alpha helix, beta sheet and secondary structure come, beta sheet conformation come, and after this area is also a slightly allowed region. There are slightly van der Waals radius are present. So from this, you can see from this modeled structure, all the residue which are coming in allowed region or not. From for this structure, you can see all the residue are coming in the allowed region, allowed and favored region. So you can further go ahead with this modeled structure. Below that, there is mol probability result also. Here you can see the Ramachandran favored region, that is showing 97.84 residue are lying on the favored region. These are colored region are all the favored region, and all the residue are lying on the favored region. And these are the same result which you got in the previous page, cumin disco score. These are showing the flexibility of the residue, like how flexible your residue while the 3D structure is modeled. That is same the cumin score. And here you can see the cumin score with the template and the target, and you can see the 100% coverage except for some residue from the N terminal and C terminal. So after this, like all this parameter you checked, and you can select your model structure, you downloaded the model structure. Now you can see what other type of analysis you can perform. Like I show, like this is the same structure we model. You can open this structure in the any of the visualizing tool. Here these are the model structure which we built from the Swiss model. So you check the Ramachandran plot and other parameters to check the quality of the model. Now there are other also software tool which you can further use for the model assessment, because like in bioinformatics, before homology modeling you do the multiple sequence alignment, and then the homology modeling you build the you build the structure of your unknown sequence. Now you have the structure is the functional unit. Now what types of analysis further you can perform? You can perform the docking. You can see, because you can't perform the docking with sequence, that's why you need the structure. Now you have the structure, you can perform the docking. You can further see the molecular dynamic simulation. So all the other analysis and good analysis you can perform after this step. So this is the very basic step for the further good step. So this should be accurate. That's why when you are building the model using the homology modeling approach or other approach, like for the modeling, homology modeling is the we are using the web server, and they are a standalone tool also. So if you are not able to use the standalone tool, so you have to further recheck your builded structure. So now we just use the Swiss model to recheck our build structure. So in the further next session, or I think, I don't know what is time left, or I can show what types of other analysis we can do for the builded structure, so that we will be 100% sure that our model structure is accurate, and we can further continue with the other types of analysis. Okay, this already done. Yes. I prepared the other types of analysis for the next session. So if you guys all also want to perform this step which I performed in front of you, and you have any query or ask something, we can perform equally. So you will get you will also have the data, and we will do the other types of analysis for the next session. I'm not able to hear you all. I don't know. I think I'm
Hello,
not
Dr.
able to
Poonam.
hear.
Hello, Dr. Poonam. Can you hear us?
I will rejoin, I think. Then I can hear you.
Hello.
Dr. Ravi, ma'am is rejoining. Then probably she'll be able to hear us, sir.
Right.
Okay, in the meantime, participants, we have some very important announcements for you all. First of all, please join the next session sharp at 2. Our next session is from 2 to 3. Then again, we have another session 3 to 4. Hello, ma'am. Ma'am, can you hear us,
Yeah,
Poonam ma'am?
yeah. Now I can hear you. Earlier,
Okay,
I
ma'am.
can't hear you any often.
Okay, ma'am. Okay, ma'am. Ravi, sir, you may continue with the session.
No, you convey the message, then we'll resume.
Okay, okay. From 4 o'clock onwards, we are having a valedictory session and it will be on a different link. So the link has already been emailed to you. Still, we'll be sending it to you via WhatsApp and email again. So kindly do join the valedictory session from that different link only. Okay.
Okay.
Uh, Ma'am, sir, you may please continue.
So first of all, I would like to convey my thanks to Dr. Poonam for such a wonderful session on homology modeling. And if we talk about the drug designing, then we can say that we must start with homology modeling if we do not have any crystallographic structure of receptors. So this is very important topics and very well explained by Dr. Poonam. And maybe in the next session, post lunch session from 3 to 4, she will includes more experiment on a structure validation and something else. So, but few of the questions from participant, Dr. Poonam, you may check in the chat box also, that the one question from Abhishek Char, ma'am, what should be the optimum range of choosing resolution? I think he is
Okay.
asking about when we choose a
X-ray
template.
crystallography. Yeah.
Yeah,
Yeah,
template,
it's
right.
the very good question. Like along with the coverage and identity, you should consider the resolution. So the maximum resolution you can select up to 2.9, not after 2.9. You can 2.9 is also the worst condition where you are not having anything and coverage also, but you can consider up to 2.9.
That's great. And another question from Indira. Indira, that ma'am, what is the standard range for cumin disco?
0 to 1.
I think 0 to 1 is good.
Yeah.
And but one thing that which one the good toward the 0 or toward the 1?
I think less is good. Less is good.
I mean, toward 0 is the good, right?
Yeah. Up to less
Okay.
is good.
Okay. And next question from Aditi Nag. That is, ma'am, I have a predicted protein sequence from a genome sequencing data, a non-PDB sequence, right? So will I need to do some additional step for modeling? I mean, what I am getting from this statement that she is having some unique sequence, maybe some
Hmm.
genomic sequence that is not
Yeah.
available on PDB. I mean, the structure is not available on PDB. So she want to know that whether she need to add some step before
Yeah.
modeling.
Actually, yes, like for the protein structure prediction, there are different approach. So what we are discussing today is homology modeling, and this is the knowledge-based approach. Knowledge-based approach. Why we are telling this as a knowledge-based approach? Because we are having something in our database, like we are looking for the sequence similarity. So for homology modeling also, sequence similarity, if you are having sequence similarity up to 30%, then only you can use this knowledge based approach. But what is your question? If you don't have the PDB structure in the database, then how you can model your structure? For that case, you have different approach, like threading and the ab initio approach. So in that case, you can use that to approach, because this is not the ideal case. Like you will always have the structure similarity with your unknown, with your target sequence for which you have to model the structure. So there are different approach also, and in that case you can use those approach.
Okay, the same question added to Dr. Aditi that, ma'am, the sequence I have is non-reported, but the protein I suspect is well reported in other species. I mean, protein is reported in other species, but not in the same species. So I think for modeling, we
Yeah.
can use as a template from other species.
Yes.
What do you think, Dr.
Yes.
Poonam?
Yeah, you can use template. You are, yes, what is your question? If species is not a problem while you are building the protein three-dimensional structure, that's why we are using different criteria. If it's from other species also, you can see the coverage, coverage range, how much coverage it's covering. So you can use those things.
Right. So thank you, Dr. Poonam. We will resume our hands-on training session from 3 to 4. And before post-lunch hands-on session, we have one very important lecture from 2 to 3. So I request participants to join that lecture and continue with the hands-on session also. With the same joining link, right? And for validatory, we have different joining link that you will receive in your mail and WhatsApp group also, and probably we will resend it, right? So thank you, Dr. Poonam, and we'll see you in the next session from 3
Thank you, sir.
today. Thank you.
Our speaker for the session is here. We are just waiting for the participants to join in. So if you know anyone who's a co-participant and is your friend or colleague, please request them to join quickly.
Hello, Dr. Jeenat. Welcome to the session.
Yeah, hello, good afternoon.
Yeah, good afternoon. So we will start this session very soon. Few of the participants do not join yet, so just we are waiting for them, and hardly 3 to 4 minutes we will wait for them, right?
Yeah, okay.
Although more than half of the participant has joined because
It's lunch.
lunch.
Mhm.
Welcome, Zenat ma'am.
Yeah,
It's so nice to see you after such a long time.
yeah, same here. Should I start my like screen sharing now, or should I wait further?
No, we'll wait. We'll wait for the participants.
Yeah, it's okay.
It's okay.
Ma'am, what time it is there?
It is 11:32 here in the morning. Like I'm in Jeddah, like Saudi Arabia. Like it's a weekend for me today.
Okay, Friday, that's why.
Yeah, being Friday, it's a weekend.
So you have 3 days long weekend there. I mean,
No,
Friday,
just
Saturday.
no, it's just 2 days, Friday and Saturday only. Sunday
Right.
is a working day for me,
Oh, right.
unlike you all. So I think you all are also faculty like Manali there in Amity.
Right.
Like you all are colleagues of Manali. Okay.
Yes, ma'am.
Is Sanket also there?
Sanket sir is also here, but he's not in right now an active
Yeah,
part of the workshop.
He's my junior in, like he was my junior in AIMS.
I just told them
Mm-hmm.
he's still working in the field of X-ray crystallography only.
Yeah.
We have deviated from there, but he's still on it.
Yeah, it's the best thing to be like to continue as your doctorate. Like it's the best thing.
We'll just give 5 more minutes, then
Yeah.
we'll start with the session.
Yes. Okay. Do you have any background noise from my side or is clear?
Absolutely clear. We won't be fishing any internet issues, don't worry.
Yeah, okay.
Manali ma'am, I'm just sharing the IEEE background for
Yeah.
clicking a pic.
Okay.
Manali ma'am, we can begin the session.
Okay.
Can
Good
I do-
afternoon. Yeah. Good
Who's the-
afternoon, everyone. Welcome to the second part of the second day with a very interesting session. We have with us Dr. Zeenat Mirza, who is currently working as an associate professor in King Fahd Medical Research Center, King Abdul Aziz University. She is a very active part of the bioinformatics unit and fundamental applied biology research group. She has previously worked as assistant professor in Department of Bioinformatics, Jamia Hamdard, New Delhi, India. And her PhD is in biophysics from All India Institute of Medical Sciences, New Delhi, after a dual master's degree in biotechnology and bioinformatics from Jamia Millia Islamia, New Delhi. She has worked on various diseases like Alzheimer's, on target proteins like A-beta peptide, on venom proteins and few other proteins in synergy, and gained experience in protein purification, X-ray crystallography based structure determination, proteomics, and bioinformatics analysis. She has 5 structures deposited in RCSB's PDB. Her present interests lie in structure-based drug discovery, proteomics, bioinformatics, functional genomics, and cancer genomics. She is currently involved in many research projects and published more than 64 research articles. Without wasting any further more time, I'll request Dr. Zeenat Mirza to take the dais and inform us and enlighten us on an important topic. Thank you, ma'am. Zeenat, ma'am, you're not audible. Unmute yourself, please. Yeah.
Okay. Hello, everyone. A very warm good afternoon to all of you. I will start my screen sharing now. Is it visible?
Yes, ma'am, it's visible. May I request for, yeah, the full screen is fine. Thank you.
Okay. So the topic which I choose for today's presentation is identification of disease biomarkers using a system biology approach, like the holistic identification, where we get a bird's eye view to complete a story, like what's the scene at the genomic level, what's the scene at the transcriptomic level, and what's the scene at the proteomic level, to find diagnostic biomarkers or prognostic biomarkers, and the most important, therapeutic biomarkers. My topic will be like briefly about first I will start with the brief background, followed by the data analysis of microarray based platform like data, which is known as transcriptomic data analysis. I will discuss about the in-house data as well as the data from the public domain, which we do the meta-analysis for disease correlation, where we can correlate one or two or more diseases to find common biomarkers, and for novel biomarker discovery. It can be one gene protein, or it can be a gene signature, like few genes together, which are characteristic of a particular disease. The later part of my talk, I will be talking about single nucleotide variation annotation. So the basic theme, like I will start, what is a disease? Disease is any normal deviation, like any harmful deviation from the normal state. So we should know what is the normal healthy individual state of an organism and what happens when the organism is having a disease. Disease is characterized by certain signs and symptoms, but signs and symptoms is something which is quite vague. Like it can be fever, it can be like diarrhea, it can be chills, it can be like what's the blood pressure or the palpitation. So these are something which is generalized, like the doctor sees the eye color or the skin color and say that, okay, this disease, the person is suffering from this particular disease. And the biomarkers, which are biological measures of a disease, are objective. They tell exactly if this gene or this protein is upregulated, that this is characteristic of this particular disease, or if at a certain time the genes are down-regulated, underexpressed in a particular disease. This is the best way to characterize the disease in the present times. And the biomarkers are the biological measures of a biological state. The best definition of a biomarker is that it is a characteristic that is objectively measured and evaluated as an indicator of normal biological processes, pathogenetic process, or the pharmacological response to a therapeutic intervention. The disease biomarkers are broadly classified into 4 domains, like 4 areas. The basic being the physiological one, which includes like blood pressure, the pulse rate, the body temperature. It can include histological ones, where we talk about the histochemistry, the immunohistochemistry of the patient's like tissues taken and the like biopsy is done, and the slides are made and then visualized under a microscope for certain proteins presence, like for P53 or for estrogen receptor and all. That is the most traditional way of categorizing the disease, especially cancer. The third being the radiographic methods, like which include imaging techniques. It can be like X-rays, CT scanning, MRI. The most important, which is the focus of my talk today, is the molecular biomarkers. These are non-imaging biomarkers, which are generally gene or protein, which are either upregulated, most commonly upregulated one we talk about, sometimes underexpressed, the downregulated ones also. And these, the study of these disease helps in the personalized or precision medicine for individually tailoring drug therapies or for having prognostic biomarkers for the disease, like the course of the disease, to follow the course of the disease during the treatment of the patient. The molecular biomarkers I'm broadly classifying into 3 areas. The diagnostic one, to diagnose the disease. It detects or confirms the presence of the disease or condition of interest in an individual. The prognostic biomarker, it tells about the likely course of the disease, like the outcome of the patient with the therapy being given with the patient. And the most important being the therapeutic biomarker, where we design against the drug target, whether the gene or protein, which is a drug target, and we design rational-based drug design is done for that particular drug target. There's also one where we combine therapeutic and diagnostic together, known as theranostic biomarker. Okay, now presently, like before, like 50 years back or so, or when the science evolved, like each science was being studied separately, one science one at a time. But today, the modern way of studying science is to combine different aspects of science together, like different omic science, like the genomics, it's combined with proteomics, it's combined with transcriptomics, even with metabolomics, lipidomics, glycomics, together to give a full picture of the body, a full picture of the cell at a particular time. And because nothing is working in isolation in a cell or in a biological system, nothing works in isolation. Everything is like different interplays are going on. And studying one thing only doesn't give the complete picture. So we have to combine different omic platforms. Even now, the computational biology is very well combined, like machine learning, the artificial intelligence, because we have big data, huge data is there, and it needs a strong computational power, for especially for the high throughput data, which is generated from like from microarray platform or NGS platform. Like we need a high throughput like data, we need a strong computational background also. And different science like the molecular biology, biotechnology, biochemistry, biophysics, everything is combined at this level. So initially it was one gene approach, individual, one gene, one protein at a time approach. But now it is like holistic approach where we combine all gene together, all exome together, like it's like all proteome together for the holistic system biology study. And because there's a huge growth, exponential growth of the data being generated from these platforms, the multidisciplinary bioinformatic approach is important to analyze, to do data mining, to better understand and interpret the complexity of a disease. So you all know that bioinformatics is a hybrid science where we combine information technology methods, developments in IT to analyze the biological data, where we develop software as well as we use those softwares for better data analysis and interpretation. So this is the most basic theme, like the gene codes for the mRNA, the transcripts, and then transcripts codes for the proteins. The DNA is the static entity. It's same throughout your life, except when there in certain conditions, there is an induced mutation in the lifetime of an organism. Transcriptomic is the mRNA being like coded from the DNA, and the proteins are the actual player molecules, which are the enzymes or your like transporter molecules, the molecules which are mainly involved for the phenotypic display of the person or the disease. So genotype remains same, it's like static, but proteomics is a dynamic entity. The proteome is a dynamic entity. And also the most important thing is that the sequence determines the structure and structure determines the function. Like if there is any change in the sequence of a gene or a protein, it will somehow reflect in the structure of the gene or protein. It may or may not be a strong effect, but it will be there. And the function will also vary based on the changes in the structure induced by the variations or the SNPs or the mutations induced in the gene sequence. And we also know that studying genome is somehow simpler because of so many advancements done in the high throughput technology for the genomic platform. But studying proteome is much, much more difficult. And the Human Genome Project is completed like 20 years back, it's completed. And we know the exact number of genes in an organism, in human being. And it's around 25,000 genes, which is widely like quoted. But the number of proteins in an individual, number of unique proteins in an individual is much, much higher. Other than that, the abundance of, because our genome is fixed, like we know that one is from maternal side, one is from paternal side, it's fixed. But the proteome, it's like the abundance of proteins is much, much higher. There are so many proteins which have so many variants, and this arises because of differential splicing. And the abundance of protein varies to an order of 10 to the power 12 or 10 to the power 10 like magnitude. So studying protein is much more difficult because it's much more complexity is there. And this I already talked about, that what is proteomic is the identification, characterization, and quantification of all the proteins involved in a particular pathway, cell organelle, cell, tissue, organ, or organism. to provide a comprehensive data about that organism or that system at one time. The techniques for sequencing is like NGS, or the first, like previously Sanger sequencing was done, but now the whole exome sequencing is done using next generation sequencing platforms. For transcriptomic studies, we commonly use microarray along with RT-PCR. And for proteomics, the studies is done by 2D gel electrophoresis and mass spec. The techniques of genomic and transcriptomic is very well advanced, but the techniques for studying proteomics is not so advanced because the whole proteome has not been well completed till now. It's for us, the proteomic technology suffers from some limitations. First being the limited throughput, the low throughput of proteomic platform. Second, there is no absolute quantitation method for proteins. Third, like for doing anything in genomic, we do PCR. We can make multiple copies of a particular gene, but for protein, till now, we don't have any technology like that available. So for studying protein, it's like only when it can be done only when we isolate it or purify it from a natural source. So another way to study proteome is to study the mRNA level, study the transcript level, because proteins will be formed only from those transcripts only which is formed in the body. So measuring the mRNA level gives an idea about the over and the underexpressed proteins, or the upregulated pathways or the downregulated pathways. So transcriptomics is an insufficient but a shortcut way to study the most functional aspects of genomics. And this is commonly done, like previously it was done by like for single gene at a time, like northern blotting was done, or qPCR is done for one gene at a time. For multiple genes together, we do cDNA or array or oligonucleotide array, which is commonly known as microarray, where the gene probes are attached to a chip and the experiment is done. So, and another way is to do RNA-seq using high throughput NGS to record all transcripts and how many copies of that transcript is there, to know the abundance and the number of copies being made. I will discuss further about the microarray. Like overall, this helps in this meta-analysis. And by meta-analysis, I mean the data analysis of those transcriptomic data, which is available in the public domain and the in-house also. Because for us, we have a good platform of Affymetrix and Agilent microarray platforms are there, and we do in-house microarray regularly. But suppose we don't have in-house platform, anyone interested in these kind of study can take the data from public domain, from NIH domain, and they can just do meta-analysis using the softwares and give some, like you can interpret so many like good conclusions from the data already submitted in the databases, to explore the molecular basis of the disease, to find the differentially expressed genes, whether it's upregulated or downregulated. Also, most interesting, like we can find correlation between two or more diseases, like between Alzheimer's disease and Parkinson's, between like different type of cancer, we can find some correlation. A common biomarkers can be there, and for especially and for sure biomarker discovery. And when we get some good biomarker, we can propose them as potential diagnostic biomarker or prognostic biomarker, and also as therapeutic biomarkers, where we can design drugs using the rational structure-based drug design approach. So this is the workflow of a microarray experiment. I'm not going into the wet lab work, like the work done by the technicians in the lab. I'm not going deep into that, but just to give an idea, we collect samples, tissue samples, or sometimes biopsy like samples from the patient, like for example, cancer as well as healthy control, and we do the microarray experiment. After the experiment is over, we scan the chip, and the raw image is somehow like something that like I'm showing here as the raw image, but you cannot deduce something from just the image visualization. So lots of statistical work is done by the software. We here use Partech Genomic Suite to clean the data, where we do pre-processing, normalization, and many statistical like filters are applied there to find the fold change. By fold change, I mean what is the basal level and how much upregulated the gene is there, like 2 times more, 3 times more, or under expression. We also find the under expression, like the housekeeping genes and the expression of the genes, other genes which we are interested for us, because some pathways or some genes get suppressed during a particular disease. And then the pattern recognition is done, and we take the primary data, analyze and interpret, and this is done using the bioinformatics software, the bioinformatics suite, which turns our data into discovery. this is another important flow chart, like where we, after the data generated, we import the sample into the Partech genomic suite. This is the spreadsheet in the Partech suite where we import the data. Then we do some quality assurance and quality control, and we get PCA map, like scattered plot is generated using principal component analysis. And then where it's like puts the spot, like each sphere is like about one case sample, one case. And similar type of cases grouped together in a one ellipsoidal, like you can group into one ellipsoid, which I'll show you later. And differentially expressed genes are tabulated as a table with p-value and fold change. And ANOVA is applied, which is the variant, like we calculate the variance between the sample and within the sample. We calculate the difference between the variance in within the sample and within, like between and within. And then we do we construct heat map, which is by hierarchical clustering of significant genes. If we lower the p-value cutoff, the number of significant genes reduces, but we cannot lower it too much because sometimes the important biomarkers will be lost by that process. So it has to be like, we have to be very careful about the stringency of the cutoffs. So now the first step is the data retrieval, either from in-house data where we have .cel files, and it's widely accepted in scientific community that like whenever we complete a transcriptomic platform data, like whenever we complete a transcriptomic platform analysis, we have to submit into the NCBI's GEO site before publishing that data. And so in NCBI GEO site, you can have every world over scientific community data is already there, and is free for like downloading, and anyone can download and analyze or reanalyze it multiple times from different aspects, different perspectives to find novel biomarkers. And then data sets having common platform are merged to minimize variation and obtain bigger cohort size. We also do meta-analysis sometimes along with in-house data to increase the cohort size, because suppose we have just 10 samples of a particular cancer, suppose ovarian cancer, and 5 control. This is very small sample size to have some statistical significance. So it's a good way to add data from the GSC side, from the GEO side, and then increase our cohort size for a much better statistically significant interpretation and like results. And this is the homepage of the Partech genomic suite, where we just keep on importing the data and to start the analysis. And then later we find the common regulatory networks, and this helps determine the common features of one disease or 2 or more diseases which are being compared. So the .cl file is imported, and a log transformation is done using robust multi-array average algorithm. Normalization is done. PCA plot is done to find the variance between sample groups. And usually we keep a false discovery rate cutoff and like around and a p-value of 0.05 and 1 to 5 to 2-fold change, like fold cutoff change. And then average linkage hierarchical clustering is done to generate heat maps. This is the PCA plot. Like here, suppose like the green spheres represents the normal case. The red one are the disease cases, and the blue one is the control. The microarray platform, they also provide some of their own control chips, and these are for the control. So we can see like the green and the red are from the Arab population. So they have some similarity, but we can clearly distinguish this ellipsoid with the disease in ellipsoid. Similarly here, the disease one is blue and the normal control is the red one. So they somehow lie together in a plane, in an ellipsoidal plane. Similarly here, the blue one is the breast cancer type and the green one is normal. And it's okay to find 1 or 2 outliers, which are little but they still lie with that plane. Then heat map is constructed. On one axis, we have the samples, and the other axis is the gene sets. And the high expression is given by red, and the medium one at white, and the lower expression, the under regulated ones are shown in blue here. But this color coding is not stringent. You can have colors as per your own choice. And this is of our own data, where I'm showing the difference between the normal versus cancer. The whole blue area is the genes which are getting down regulated, and the red ones showing the upregulation one. We also have some samples which are like down-regulated here. Here I'm showing the comparison of breast cancer tissue versus a normal tissue. And we also sometimes categorize our data based on like which stage of cancer it is, stage 1 cancer, stage 2 cancer, or stage 3 cancer, or on base of other histological parameters like estrogen receptor positive or estrogen receptor negative, or HER2 positive or HER2 negative. These are already known clinical biomarkers for staging of breast cancer, for diagnosing as well as staging of breast cancer. And we get like map like this. Here also I'm showing like the same for breast cancer data. The right side one is the normal, upregulated and downregulated one. Then grade 1, we had just 3 cases. So you can see on the x-axis at the bottom of the screen, like 1, 2, and 3 cases were only for stage grade 1 cancer, and the rest grade 2 and grade 3. And this also shows that usually we get grade 1 cancer, like very limited cases come in the hospital for grade 1 cancer, because cancer usually diagnosed only at a later when this cancer has progressed further. And then grade 2 and grade 3, and this is the hierarchical like clustering of the genes based on the grading of the stage of the cancer. Similarly, here is like when the cases are ER negative. We had 3 control cases in this data, and we compared them with the ER positive as well as ER negative cases. Okay, so this was the heat map, the hierarchical clustering. Then we make like Venn diagram. That's easy, like mathematical Venn diagram. The software makes this for us very easy to do. It like here in the left one, I'm showing the stage 1 breast cancer versus the normal. By 0, I mean the basal, the healthy individual, the healthy control. So this is the stage 1 with 0, stage 2 with the normal, and stage 3 with normal. And the middle one, the 429 genes were common in all the 3. Like when we compare the 3 stages of breast cancer, 429 genes or differentially expressed genes were common. In the second figure, I'm showing where I have combined which I have done meta-analysis of the data in public domain for breast cancer itself, where I have downloaded several data series from NCBI GEO site, and then I found these 25 differentially expressed genes which were of interest because these genes were upregulated in all the 4 data series. So in that work, I proceeded further with these 25 genes. This is the table showing the gene expression and the genes which are upregulated in our breast cancer data. And these genes were upregulated, but certain of these genes were not having any association with breast cancer when we did the literature search as well as database search for the significance of these diseases in the cancer. But certain genes like CXCL10, topoisomerase 2A, the histone proteins, or these proteins were having a good significance in breast cancer. So we can stay further that these genes are upregulated in cancer. So these can be used as a diagnostic biomarker, or it can be used to design drugs against them. These were the genes which are underexpressed or downregulated in breast cancer in our data. See, sometimes some genes are underexpressed, like minus 45 times. It's too underexpressed, or the expression has been decreased tremendously for this particular gene in breast cancer. So I'm just trying to give you a flavor of the results which we get after the microarray analysis. And similarly, up and down regulated genes in different stages. Like the grade 1, grade 2, and grade 3. So by this differential tabulation, we can have some distinct biomarkers for identifying each grade, or categorizing each grade of the patients, or each group of patients based on the grade. And because there are specific therapies for each staging of the cancer. So this translational research helps to correlate the omics platform data to find biomarkers for staging, for prognosis, to predict recurrence or for therapeutic relevance. And we assume here that the statistical significance is also true and equivalent to the biological significance. And I'm showing here the 4 most commonly known, proven and validated biomarkers for human breast cancer, which is estrogen receptor, CD34, P53, the tumor suppressor gene, and HER2. So the next step after this, finding the differentially expressed gene, is to put these genes into the pathway analysis software. We commonly use here Ingenuity pathway analysis, which is like one of the most respected or very well, it is a huge database. It has a huge database to find correlation between the differentially expressed or differentially regulated genes. So we import those genes, and the software itself makes predicts a pathway where those genes have some role to play in a common canonical pathway. And we do this to find the disease etiology, to understand the disease etiology. How does the disease start and how does it progresses? And we need to assign, because we get a huge list of differentially expressed gene, and we need to assign function to each gene, like what kind of function it performs, where is it located, which biological processes is involved in. And till now, only 5% of the known genes have assigned functions. And without understanding the complete function performed by a particular gene, drug discovery cannot be done. We should know the function of the gene, then only we can plan to block or inhibit that particular gene or protein. So we need to understand the pathway very well to intervene therapeutically in the disease process. So I already talked about this, we do pathway analysis to find the function of the genes, to find the biological processes they are involved in, to see which pathways are dysregulated or disturbed in the particular disease mechanism. And whatever I am talking about is not specific to a particular disease, but is very generic. It can be for any kind of disease. It can be for cancer, it can be for neurodegenerative diseases, it can be for cardiovascular, any metabolic disease, or wherever gene have a role to play. It's something very generalized. And when we do differential, when we find the initial list of differentially expressed gene is usually in thousands. But when we reduce it to a pathway, only tens or 20, 2-digit, we get a 2-digit number of proteins or genes which are playing the important role in the significant biological pathway. So pathway analysis helps to increase the statistical power by reducing multiple hypotheses. Because when we just have a gene list, it can be involved in, those genes can be involved in plethora of pathways, different independent pathways. But when we combine and see where do they fit into a, how many genes fit into a particular pathway, it makes more sense. It's it gives a direction to the analysis being done. And it helps to identify the molecular signatures, transcriptomic signatures, where not only one, but a panel of genes, 5-6 genes are combined together for diagnosing or categorizing the disease stage or the progress of the disease. Some tools for pathway analysis which are commonly available and being used are David, IPA, the Ingenuity pathway, which I commonly use, Pathway Studio, KEGG, and Wiki pathway, and so many. Most of them are free, but IPA is a commercial software. This is the homepage of David. Okay, and there's something there's a difference between pathway and a network. Pathway is a linear flow chart, it's a linear sequential flow, it's a 2-dimensional presentation of the data. But whereas at a network, it's a multi-dimensional data, where multi-dimensional genes and proteins are playing a role. So, and what do we mean by node and edge? Nodes are the connections. It can be a direct connection or it can be an indirect connection. Usually by a straight line, a solid line is for direct connection, and a dotted line with gaps is for an indirect connection. And the pathway we connect, will have a list of interconnected nodes, and the nodes they symbolize a particular gene or a protein. And in a network, there are many genes and proteins together from every direction coming, it's an interconnected pathways, forms a network. I'm showing an example, this is the left one is the pathway, the right one is a network for EGFR. This is a KEGG pathway, like pathway for KEGG for cell cycle, and KEGG is free and it can be used. This is like about IPA, the Ingenuity Pathway Analysis from Kaizen. Kaizen, like the IPA is a very, like it's a robust platform where we just have to put our differentially expressed gene list, import there, and it predicts the best available pathways for taking care of the maximum number of genes, which we found in our results. It gives about the top networks, it gives the top canonical pathways, and the diseases where the genes are mostly responsible for, the molecular and cellular functions like cell cycle, cellular movement, and the physiological system development and physiological, what is the physiological system level, like it's cardiovascular, more involved in cardiovascular or organism development, like that. Like here, I'm showing like our results, the breast cancer biomarker, which were total combined for, there are some which are for diagnosis, like diagnostic biomarkers, some are for prognostic, some are for therapeutic biomarkers combined together as a chart. And here I've like separated from this like whole like figure, I have separated into three. This is like the diagnostic ones. Like we can see the upregulated ones like MMP11, matrix metalloproteinases, or aura kinase, or like CXCL9. These are upregulated in breast cancer. The green ones are underexpressed. And the intensity, the color intensity, like it's the more overexpressed is more darker in shade, the less overexpressed lighter in shade, something like that. And these are the prognostic biomarkers. Like BRCA2 is one of the most common used biomarker for breast cancer screening also. And there are many more, like these are involved in like cell DNA repair or something of the organism. And these are the therapeutic biomarkers. These are the progression biomarkers, and these are the therapeutic biomarkers. There's some biomarkers which are the opposite of it, which are the safety biomarkers. Like when we start the therapeutic intervention, these biomarkers are commonly disturbed. So we should take care and keep close monitoring of these biomarkers. This is a like table of our published data for our own published data for renal cell carcinoma, where I'm just showing you the table of the differentially expressed gene. I'm showing you like a screenshot of the upregulated genes with the p-value and the fold chain, and the underexpressed, the downregulated genes. Like we can see the -4 like values in the fold chain for renal cell carcinoma. Like in this case, we selected cyclin D1, and we further work more on this particular protein in this publication. These are some of the pathways like made by IPA. Here I'm showing like leptin pathway. Listen, the leptin network. The red ones are the upregulated ones. The green ones are the downregulated ones. Here is the leptin molecule. So it's very clear, like this is the cell. This is cell membrane, and this is the nuclear membrane. The proteins are the different enzyme kinases, the membrane transporters. Like we can see so many, and we can see some direct pathways, like the solid lines, or the indirect pathways, where there might be more molecules in between. And this is the right side, the network I'm showing you, the network for thyroid carcinoma. And here also, like it's a whole set of genes and proteins play an the interplay. And we have so many relationship, direct as well as indirect, we can see between different kinases and different cell membrane proteins. This is just like a key, or the legend key for the IPA, what determines like what is like kinases is usually shaped as a triangle, or growth factors are shaped like this, or cytokines like this, and direct indirect relationships. Here I'm showing Wnt catenin signaling in renal cell carcinoma. This is also from our in-house data, where overexpressed and underexpressed are being shown. The underexpressed were few, like the green ones, and the overexpressed, like here in the blue, in the red. Okay, the next step is, because till now we were telling about the differentially expressed genes and their network and their pathway. So the next step logically is to study these proteins, or these gene products into a protein network. And the most common platform for studying protein network is string. We just have to import just have to enter the gene list, and the string makes the possible prediction like of the pathway of the network, actually, not the pathway, but the network of the functionally significant interacting proteins. This is the interaction network for leptin protein, which I just discussed before. And string is absolutely free. So gene set enrichment analysis is done to know the gene ontology, to give a particular function to each gene, to functionally categorize significant genes, and to link whether that the gene is linked with the disease of interest, yes or no. So it tells about the molecular function, like what are the genes involved and what function does it perform. Cellular component, where does it located? Like, is it like cytosolic or nuclear? Which biological processes it's participating in? So the gene ontology, the GO, is a structured hierarchical vocabulary to describe the gene function in these three broad areas: molecular function, cellular component, and the biological process. And this is a generalized table for gene ontology, where the genes which we imported are most of them are falling in oncogenesis and apoptosis. And this IP actually is based from literature, already published data by the world over literature, and they do data mining from there, from the published literature. So it categorizes the gene of interest into the major function being performed. So to conclude the transcriptomic slides, I would say that the genome-wide profiling, the transcriptomic profiling followed by pathway analysis helps to identify disease signatures or molecular biomarkers and categorize the role of specific gene products that use the protein with clinical parameters, and attempt to identify biological function, expression signatures, and the disturbed pathways in a specific disease serve as a novel targets for diagnostic and prognostic biomarkers and potential therapies. Because there's a huge data, the exponential data, the high throughput data, platforms, they generate large data. So, and we have a lot of variation of protocols, platforms, because different gene chips have little variation among themselves. Different platform like Agilent platform or Affymetrix platform, they have little variation. So it has to be statistically combined together for analysis, and it needs a system biology approach using lots of improved software, bioinformatic based software. So the next step after having potential therapeutic biomarkers in hand, we design drugs against them. And the drugs should be which perform the desired function, they inhibits the activity of that upregulated genes. And the best drugs are those which are given at the lowest dose and with fewer side effects. So rational drug designing comes into play. And this is just a flow line of how to identify when the disease is identified. We do the holistic system biology approach of genomic, proteomics, and metabolomics to predict the potential targets or biomarkers for, especially for personalized therapy or the precision medicine. And then high throughput screening is done for thousands of compounds, and we can get the best available compound for inhibiting that particular drug target. And the testing is further done, and lots of molecular modeling and simulation can be done to improve the activity. So the next step, this completes my transcriptomic data description. Now I'm going to another part of my talk, which is the single nucleotide variation annotation. This is the genes, the genome, human genome. We all know that we all share our genome, almost at maximum of the genome is same for each individual, for each population, but there are slight single nucleotide variation or polymorphism existing amongst ourselves, between the population, between different Caucasian populations, different from Arab population, which is different from the Asian population, based on those SNVs. And also in the case of disease, sometimes a single nucleotide variation occurring in a gene, which is not a major mutation, but it's just a single nucleotide variation. It changes the effect to a drug. It changes the course of the therapy. It has a quite reflection on the phenotypic of an individual, phenotype of an individual. So SNV annotation is must to predict the functional effect of a gene. And we know that the real reflection of a disease phenotype is seen at the protein structural level only, because the mutation or the variation is at the genomic level, but the structural and functional consequences can be seen at the protein level only. And these are just single amino acid substitution, and they affect protein function by altering the shape, the shape of the protein somehow. And it can be, usually it changes the folding of the polypeptide chain that increases or decreases the stability. Usually it decreases the stability of the folded conformation. And it also sometimes affects the post-translational modification site, because if a protein, the post-translational modification site is changed, that PTM cannot be done. That changes whether the protein will be like taken away from the cell after the performing the function or not. Like it's like a flagging of like the post-translational modification is like the flagging of the protein like landscape. But if it's changed somehow, the next downstream pathways get disturbed. The single nucleotide variation can also happen at the catalytic binding site, at the ligand binding site, which will change the catalysis of that enzyme, especially. It can also change the interaction, because nothing is working in isolation, and then two proteins have to interact with each other. They physically interact with each other in the pathways. So if the common binding domains are disturbed because of this any SNV which is like induced, the interaction between two proteins changes. So that will cause an effect on the pathways. I'm just showing like what is an SNP, like it's just a single nucleotide variation. It can happen in non-coding region. It can happen in the coding region as well. In fact, we know that 98% of the human DNA is junk, and it's kind of a way of nature to protect our genome from random mutation or variation happening. So most of the variation will be there in in the non-coding, the junk DNA side, which we don't like. It's of significance for sure, but we don't know till now what is the function of the junk DNA. So we don't bother about that much. But then in the coding region also, we can have synonymous or non-synonymous. If it's synonymous, even the change in the nucleotide doesn't cause a change in the amino acid. So it's okay. It's not it doesn't have a deleterious effect. Then the non-synonymous one can be a missense or a nonsense one. When a nonsense variation occurs, it stops it truncates the protein, and the function is usually lost or tremendously affected. And commonly it's missense mutation, which we are going to talk about here. So variations in the DNA sequences can affect how we are more risk. So some individuals are at higher risk for a particular disease, and some are more at higher risk for a particular pathogen or for allergens, or sometimes we have allergy to, some people have allergy to honey, or to small things which are some drugs like aspirin, or to vaccine, another agent. And these sometimes idiotypic behavior is explained in detail by the variation in their single nucleotide variation in their genome. So we do genome-wide association study where we can have cases with disease and cases without disease, and then we compare their association, the single nucleotide variation between them, and we compare it with the traits which they are displaying. And we use the next generation sequencing platform for that. And the disease are prioritized or systematically categorized using the knowledge-based methods into 5 categories. It can be a pathogenic single nucleotide variation. It can be likely pathogenic. It can sometimes be of uncertain significance as for now, because more studies has to be done further. It can be likely predicted as likely benign or benign. So it has each SNV is based on ClinVar is a database which categorizes SNVs based on the intensity or the damage it causes, is pathogenic to benign on a scale. To understand from a protein structural point of view, we should understand the disease mechanism, the drug response, and also most importantly, the emerging drug resistance from so many anti-cancer drugs or so many antibiotics is to a great extent because of these SNVs. And to do the study, we don't need something sophisticated. We just need the data from NGS platform or from sequence data, and we structurally align the wild type, the healthy individual protein, with the mutated altered proteins to see the impact of the mutation. How much does the mutation changes the overlap? How much there's a change induced by that particular variation? And how much does that individual change causes the change in the protein stability, or it changes the binding effect, the changes the binding, or whether that SNV is located deep inside the protein or is on the surface of the protein. This pipeline of study can be applied for any disease. It doesn't matter which disease you're interested in. It can be any disease. It can be for cancer, for simple diseases, for any allergies, for any inherited genetic disorder. So we have a particular disease. We make a list of all the genes involved in that particular disease, and then we list all the SNV known in the databases for each gene. We can use databases, or we can have our own in-house and just like data. And then we check the amino acid substitution at the protein level. We have very well established databases to check this, like UniProt, ClinVar, Cosmic, or HGMD databases. And then each SNV is like further categorized based on SIFT, polyphen, I mutant, blossom, like scoring, because it's something like that. The variation from like maybe leucine to isoleucine will not be having like not a strong impact, but the variation from leucine to histidine might be having a stronger impact. They found the biochemical or biophysical property of that particular amino acid change. Or when a glycine is replaced by a histidine, because a small amino acid gets replaced by a bigger amino acid, this might affect the folding of the polypeptide chain. So so the blossom score is like seen. And like the impact of the mutation is calculated based on the variation in the two amino acids. And then the mutations are mapped on the protein structure. So this is the general workflow for SNV annotation. And like we get the 3D structure or model of the wild type as well as the mutated protein. Either we take the data from the PDB data, the X-ray crystallographic determined data from the PDB, or if data is not there, we just do homology modeling using Swiss model or ITASR or any other modeling software, Modeler or any other modeling software. And then we also do literature search or data mining, text mining for understanding the protein function, what kind of functions performs, what is the, like, which are the, where is the active site, which are the main functional domains in the protein structure, which are the known binding ligands for that protein, where are the regulatory regions. And then we see the location of the SNV of interest, where is it located? So whether it is buried deep inside the protein or is on the surface, whether it's close to the active site or is part of the active site, or is it far away from the active site of the protein, or whether it is in the ligand binding region, which I direct, because suppose it's in the ligand binding region and the SNV, there is some change induced by a particular SNV, maybe the ligand will not bind to it further. Whether it is in the ordered region or the disordered region, like in the loops, in the external loops, or whether it disrupts the interaction between two residues. Like suppose there is a cysteine which was forming in three-dimensionally, when the protein folds, it forms a disulfide linkage, and the SNV, the cysteine changes into some other amino acid, and that the disulfide linkage will be disrupted. So it will affect the folding of the protein, and it will destabilize the protein fold. It can affect post-translational like modification, and it can cause, and sometimes these SNVs are on the like protein binding domain areas. So they will cause disruption of the downstream pathways. Like for example, here, I'm showing the wild type protein. And here, like the wild type in the healthy individual, it is glycine, it changes to arginine. And this change of human glycyl tRNA synthetase enzyme. And here AMP is bound in the active site, but whereas when the mutation of the SNV, in the case of SNV, where the glycine changes to arginine, the AMP binding gets blocked, because the arginine has a bigger side chain. Glycine is the smallest amino acid, and it has just the smallest amino acid. And when it gets replaced by arginine, it blocks the path, blocks the cavity, and the AMP binding is blocked. So, and it inactivates the enzyme. Similarly, like in this, here I'm showing 4 different examples where the SNVs are mapped to the particular, like in the red box. And here, again, like glycine is changed to arginine on the surface of the protein. And it is responsible, this particular one SNV change is responsible for a neurological disease, Lou Gehrig's disease. In the second one, I'm showing the one SNV change. It causes hereditary kidney stone disease. Here, the one SNV change in the beta sheet causes the neurodegenerative disease. And in the last one, I'm showing one SNV change, like from like T to I, is in the ATP binding domain causes the resistance to imatinib drug in CML patients and leukemia patients. From these patients get resistant to CML drugs. So like second generation and third generation drugs, kinase drugs were being TKI, tyrosine kinase inhibitors are being developed for in response to these kind of SNVs. But sometimes, usually these SNVs are induced because of the stress of cancer or stress induced by the drug, which is being given to the chemotherapeutic drug, which is being given to the patient, the leukemia patients. And whoever is interested in doing SNV annotation, they can do it freely from the databases and from the software. These are set of databases which are publicly available. It's free. And we can just have a choice like gene of interest or protein of interest, download a sequence, download the list of variations already well known or reported in the databases. And we do the comparison of the SNVs analysis of what's happening in the healthy normal individual and what happens in the case of the disease SNV. And by using these softwares, Polyfen, SafeCoV, and iMutant. And here I'm showing an example of N-acetyl transferase 2, and where the phenotype changes because of the change in the genotype, change in the S1 SNV. And but the SNVs at these particular site slows down acetylation and causes atopic asthma in patients. So this was all for me from my side, and thank you.
Thank you, ma'am. A very comprehensive and exhaustive explanation of the system biology in drug development and discovery. I'm sure the participants have also realized that the mutations may also affect the designing of drugs and their effectivity. That is the one thought process I think they will be thinking about in the near future also. I think there are some questions and queries from the participants. So I'll just take one query at a time, ma'am. The first query is from Mr. Krunal Power. Which software you use for pathway analysis of miRNA?
Okay, hello, Kunal. Whatever I discussed today was just for mRNA analysis, like the messenger RNA analysis. I'm not doing miRNA analysis. There are other groups in my center which are working on miRNA platform. For me, I was using Partech Genomics Suite for mRNA transcriptomic data analysis. It's a paid software, and it's a commercial paid software, and it very well helps to and do analysis of transcriptomic data. There are some free ones also like TCGA, which can be used.
The next query is from Aditi Na. She asks, please explain the PCA again. Also, please tell us which platform is used to generate heat maps. Is it provided by the platforms like Affymetrix or NGS service provider? One thing, Aditi, the PCA explanation would take another workshop, but the rest of the questions I think may be answered by ma'am.
Yeah, the heat maps is done by hierarchical clustering. It's a statistical method, and the Partech suite itself does that plotting, and it gives us the figure. It's done by the Partech Genomics suite. It's a complete suite which does all the transcriptomic data analysis. You just have to import .cl file, and step by step it gives us the differentially expressed gene list and the heat map and the PCA plot. It is not provided by Affymetrix or NGS providers. It's separate. We have to buy it separately. And the Affymetrix or the NGS platform, the Agilent or Illumina platform, they don't provide Partech.
The next query is from Abhishek Chahar. Ma'am, as we know, antimicrobial drug resistance is a growing problem. Suppose if we were to introduce SNVs in this highly resistant microorganisms in order to change their susceptibility to drugs they are resistant to, is it possible to somehow reverse their resistance by this approach? Can we think of this approach in some way to be a successful approach to reverse AMR or solve this problem? Basically, he's asking, can antimicrobial resistance, which has been induced due to mutations in the genome, can it be reversed by introducing, I think, another mutation to overcome that AMR?
Yeah, but that approach will be a little complicated. Okay, to some extent, CRISPR-Cas9 will help there, but it's something. But what he's maybe asking is how to put SNVs into the microbes, I think.
Yes, yes.
That I think is beyond my interest and my beyond me. I don't know how to introduce those, but but categorizing or enlisting the SNVs in our drug targets, in our human proteins, where those antibiotics come and bind to. If we understand those SNVs, we can design better antibiotics which will overcome those SNVs, which can still bind to the new SNVs. Are you getting what I'm saying? The new generation of antibiotics can be designed, which are better in binding to those population. For that, it's more of personalized medicine approach, where each organism, each individual, all genes has to be known, the structure should be known, and then each SNV should be categorized. And we should be knowing okay, the patient A has this particular infectious disease, and he is resistant to this particular antibiotic. So we need to give a better antibiotic, specially designed or tailored for him, because his genes have his particular protein, which binds to that antibiotic, has this particular SNV. So we will design a better ligand, a better drug molecule for him. Are you getting
I
hope clear?
Yes, yeah.
It will be a different way. It will be a different, it will be more a precision medicine approach, a personalized therapy approach.
Yes, ma'am. I think I'll take the last question for the session. Whether Parkinson's disease and Alzheimer's disease are the result of gene level mutations only?
AD or there are so many gene mutations which are being very well listed for both Parkinson's as well as Alzheimer's disease. In fact, we have also done ourselves also, we have done some association of the two disease using microarray transcriptomic data of Parkinson's. We take the transcriptomic data of Parkinson's patient, we similarly take the data of the Alzheimer's patient, and we do an overlap of the genes to see what are the commonly dysregulated pathways in the two disease. And these mutations, there are so many genetic mutations in the patients which make them more susceptible for the disease progress in the later stage of their life. And yeah, it's true, they are genetic diseases only. Parkinson's and Alzheimer's are genetic diseases. But there are other, I think there are some environmental factors also, and some other genes which put us more at a risk for these diseases at a later stage.
I think most of the queries have been answered. I thank Dr. Zeenath again for this vast, informative lecture we had. And it was nice to hear from you after such a long time. I'm sure many of the participants would be again looking into many of the pathways now that they have come in exposure of the databases from which they can retrieve the genomic sequences and the protein SNV sequences. So yeah, I think it was pretty informative. Thank you, ma'am, again for enlightening us with this lecture. We hope to see you soon again.
Yeah, thank you. Thanks a lot.
Okay, ma'am.
All the best.
Okay, ma'am. Thank you.
Thank you so much, Zeenath ma'am. Thank you so much. And thank you for your time. And we will surely have this interaction going on in future as well, ma'am. Thank you so much for gracing the occasion, ma'am. Thank you. Yes, ma'am. Thank you so much, ma'am. Thank you.
Thank you. Bye-bye.
Okay, ma'am. See you.
Bye, ma'am.
May I request Dr. Poonam?
Hello.
Yeah. Hi, ma'am.
Hello.
We can continue with the next session of the hands-on workshop. I request Dr. Ravi Ranjan to take over the dais.
Okay, so indeed last lecture was very informative and highly correlated to the workshop title. So now welcome back, Dr. Poonam, and we will resume our technical hands-on training session. And this session is of approximately 1 hour, and we will try to wind up this session by 3:50 or 3:45 around, so that we can rejoin validatory session that will start from sharp 4 p.m. right? So what we are going to discuss in this session, Dr. Poonam?
Okay, so we will, in the last session, as we model the protein 3D structure, so we will continue with the how we can further validate those structure and what other analysis we can perform with this modeled structure.
Right. That is, I think, very important topics, because if we model some structure using in silico tools, then it must be questionable whether this structure is right or wrong, whether this structure, complete all the credential of a crystallographic structure or not, or it is permissible structure or not. So all these
Yes.
questions can be answered in this session. And the validation of a structure, I think, is one of the important part of modeling also. You can say is the next step to the modeling, but without validation, model structure cannot be passed to next experiment, right? So
Yeah.
all the best. Thank you.
Okay. Okay. Now I will share my screen. Is my screen is visible to everyone?
Yes. Dr. Poonam, you may off your camera, maybe due to that, I mean, connection issue is there. So
My screen is not visible now.
is visible now. Is visible.
Okay. Okay. Okay, so as in the last session, we modeled the protein three-dimensional structure of the unknown structure. So this 3D model, we model using the knowledge-based approach, that is the homology modeling-based approach. So we validate this 3D structure, which we generate using the Swiss model from the Swiss model server only. So we have to further validate this model structure with using the other standalone tool and the web server tool, because this is the basic for further analysis, all the analysis which you can perform with this structure. So the 3D structure is accurate or not is the basic step, and you should be 100% sure that your structure is 100% accurate, and further you can proceed with the other analysis. So here I am first discussing what types of analysis you can perform to validate your model structure, and I will show the demo also. The first thing you can do to validate your model structure is to plot the Ramachandran plot. You already plotted the Ramachandran plot from the Swiss model server, but there are other tool also, because you should be 100% sure that your structure is accurate. So you can use the different software tool based on the citation of the tool, and you can use those tools to further validate your structure. So this is the Ramachandran plot. Yeah, there are tools I can show in the demo section. So in this, you have to see all the residue which is in your protein are coming within the allowed region in the Ramachandran plot or not. Here, different color is showing different reason. The red one is the allowed region in the Ramachandran plot. So the most of the residue, if you are coming in the allowed region, that means your model is more or less correct. And there are different parameter also you have to check. So I will show you further that. And the second parameter which you have to consider is the Z score. So what is this Z score? Z score showing the overall quality of the model. In this, for this, there is online server, that is the PROSA service server and the PROSA server. And from this server, you will get this two type of graph. The first graph is showing the overall model quality. Whatever you generated the model, you can see the overall model quality. And the here in the first graph, the x-axis represent the number of residue and the y-axis represent the Z score. The more negative score is the good for your accurate model. So for this case, here the black color dot showing the model, your model is lying within this range, and this is in the negative zone. So you can consider your model more or less accurate. And in the second graph is the plot showing the local model quality. And here also The x-axis represent the sequence position and the y-axis represent the knowledge based energy. So here also, if you are having this, there you can see the two type of green bar, and the overall local quality represented by the dark green scale, and you can see for your model is lying between below the 0 and in the negative zone. So it's the local quality of the model is also accurate for your model structure. The other different types of analysis is annulia analysis, and is the tool used to assess the quality of the model protein, and by performing the energy calculation at the atomic level in the protein structure. Here you can see the graph of annulia, and lower energy value are more favorable, and negative these you can see the value are positive and negative, and the dark green color showing the value for the negative annulia, and these are the favorable residue, and there are certain residue which are with the positive value which are showing in the red color. So from this graph also you can validate your model structure. I will show you the demo also how you can do this. And the other validation you can see is the ERET. It can analyze the statics of non-bonded interaction between different atom type and plot the value of error function versus the position of 9 residue sliding window. And it's calculated by the comparison with statistics from highly refined structure. Here you can see in this graph, you can see the two horizontal line, which is the error value, that is the 99% and 95%. Reason of the structure that can be rejected by 95% of confidence level are shown with the yellow color, and reason that can be rejected at the 99% are shown with the red color. But here you can see the 95% rejection confidence value. Other types of validation you can see is the verify 3D, which determine the compatibility of atomic 3D with its own amino acid sequence, that is the 1D sequence. Here you are comparing the 3D model with the 1D by assigning the structural class based on its local environment, like local secondary structure like alpha, beta, loop, and compare the result to the good structure. So now we can see how we can perform all these types of validation. This is the tool to perform all the structure validation. What you have to do using this tool, you have to upload your model structure. So as we already modeled the homoglobulin structure, you just have to click in the choose file and you have to select the model. Now you can see your model is uploaded and you have to click on the run program. Here you can see all the validation which I already explained earlier. There is ERET, Verify 3D, What Check, Pro Check, and other. Now you have to just click on the start button for all this validation analysis for your model 3D structure. It will take some time, but you can see all those results which we already discussed. I think I already did this. Okay. After this will finish, you will get page like this. So first you have to see there was the ERET analysis. When you will click on this result, you will get this page. So ERET is showing the overall quality factor. So your model structure is 99.24% are accurate. Here you can see the ERET graph and percentage graph. This yellow line here are the 95 rejection limit. And for this, you are having only the 95 rejection limit, not the 99%. Now the second, you can check the project here. From the project, you can see the Ramachandran plot of the model 3D structure. So this is the Ramachandran plot of the model 3D structure. You can see here different color. The white is the disallowed region and these are the allowed region. When you see this graph description, you can see here all the description about the residue. Here you can see residue in the most favored region. So what are the most favored region? This red color region is the most favored region. And in your protein, there was total 142 residue. Out of 142, there are 118 residues. That is the 93.7% are coming on the allowed region. Now you can see a residue in the additional allowed region. This after this red, there is the yellow color residue. So the residue which are in additional allowed region is 7, that is 5.6%, and residue in the generous allowed region, that is the light yellow. So you hardly see the residue in the light yellow. Yes, here are some. So these are the 1.08%, and residue in the disallowed region is 0. So in your model protein case, there is no residue in the disallowed region. So this is the 100% accurate structure. You can take this structure after all this description from the Ramachandran plot. Ramachandran plot is very important validation step for any model structure. So first, whenever you will model your structure, you have to check all this parameter from the Ramachandran plot, how much residue are coming in the allowed region, disallowed region, percentage, and then you can select your template. Similarly, they are verified 3D. You have to just here you can see the result. Okay, it will take time. Okay. Okay, what verified 3D is doing? Verified 3D is comparing your 3D structure which you model with the 1D. 1D is the sequence level. So here it for you in our case is 100% of the residue have the average 3D 1D score, that is greater than equal to 0.0, and here you can see the pass. So this is also good for our model protein structure. You can see here the residue wise also average score and raw score. And yes, these are the analysis which you can perform using this web server. All these are all important analysis which you have to perform before any model structure, and among this project is the important validation step. Apart from this, you have to perform this Z-score analysis also, because it shows the overall quality of the protein. For this Z-score analysis, you can use the PROSA web server. So this is the PROSA web server, how it's look. It can show you overall quality of your model protein structure. In this also there is option where you can choose your model protein structure. You just have to click the model structure and you can open in the PROSA web servers, and there is a button to analyze. You just have to click and it will show the overall model quality and local model quality. So we can see the result for our model protein which we model for the hemoglobin. So they are from this web server you can see the 2 and 3 3 types of graph which you can use for your publication also. So in the first graph is showing the overall model quality. Here the x-axis represent the number of residue, and y-axis is the Z score, and more the negative value you are having, if your model is lying on the negative, it's good. Here also you can see the overall Z score is minus 6.69, and this is the reason where your model protein is lying in this graph. This is showing the local model quality. Here you can see this our protein is lie among this -1 something, and this is the B factor. As we already know, B factor is the very important factor to see the protein quality. So the red reason our model protein is, you can check the B factor from the X-ray crystallography structure, but in this case you are modeling your protein, so yes, with all the other validation step, you have to perform the B factor analysis also, so that you will get the quality of the model. So here you can see the red color is the less of red color here are the more flexible reason, and the blue color is the less flexible reason, and white is between red and blue. So you can see there are very little reason with the red color, so these are the less flexible, and these are the more flexible reason. Red are the more flexible and blue are the less flexible reason. So with you have the very important step for the validation is first is the Ramachandran plot, and I must say the second is the B factor, because you are modeling your protein using the homology modeling approach. And okay, so as you modeled your protein using the knowledge-based approach, and knowledge-based approach because you was having the sequence similarity. If you will not have the sequence similarity, then you can go for the threading and the ab initio approach. And so now you have your model structure. So what other types of analysis you can perform? The first, because structure is the functional unit, you can predict all the function about the protein once you have the structure of the protein. So what types of other analysis you can perform? You have the 3D structure of your unknown sequence. Now while we was doing the Swiss model, as we was getting lots of templates, so we can select top 10 template and we can perform the structure analysis. So we can get to know the common reason and important residue among those template also. So this is the first thing. Like we generally do the sequence alignment, but structure is more functionally definable than the sequence. So you must perform the structure alignment also, and you will get to know the important residue among the structure. Here, these are the structure alignment, and from a structure you can see the structure alignment, and you can see the residue which are common among all those structure. And if you will get the functional residue among any of the reason, like acidic or basic residue or salt bridge, so that you can take as a important residue for the further analysis. So this is structure alignment is also a very important step after you will get your 3D structure. And next, yes, like you can highlight, you have your structure, you can highlight the important residue. Like here, I don't know this. Okay, I will show you this alignment. Okay. So this is the alignment of structure alignment. As we know that for maintaining the overall structure of the protein, the acidic and basic residue play a very important role. And since this is the structure alignment, you can see the acidic and basic residue from the structure. So this might be the important residue for those model structure. So after a structure alignment, you can highlight those residues which are important from the alignment on the structure. So you can highlight those residue on the structure. And the third, you can further perform the docking analysis of two protein or the protein ligand. So for the online, these are the, and to perform the docking, okay, you know, you have your protein model structure. Now you want to perform the docking, like you want to know how this protein is working. So for before performing the docking, you should know the important residue. So the first approach of getting the important residue is from, might be the structure alignment, but further you have to check those residue from other tool also. So the second thing what you can do is the active site prediction. So there is the server cast-p. You can predict the active site of your model protein using this tool. So this is the compute cast-p tool. Here you just have, there is a button to calculate. It will calculate the active site. What is this active site? Active site is the site where you will have the important residue, where you can perform the docking, any type of interaction, whether it's docking or protein-protein docking, because they are too flexible and rigid docking. So for that, you should know the important residue. So this web server will calculate the important residue. So you have to just click on the calculation button, and there is an option to upload the structure. You have to click on the file, and then you just have to open the structure, model structure. You can, okay, you can see the radius, up to which radius you can want the active site. And this here you can see the email is optional. You just have to click on the submit button, and it will show you one link. You have to click on that link, and it will show the pocket. So the structure which we modeled, uh, these are the pocket here. These are the, and here you will see the important residue. These are the list of important residue from the cast-p server. So this residue, again, you can highlight on the structure using different tool. And these are the important residue present on the active site of the model protein. And these residue you can directly take for the further docking and further analysis. So here I am just showing the protein docking. That's these are the two protein, and these are the cavity. You can highlight for your work also. So this red and blue is the interacting residue. And I think this is okay, this as I am working on the COVID, so this is the COVID protein only. This is the RBD, and I am working on the nanobody, so this is the nanobody. So this is the protein interaction, and here red and blue color are showing the active site in the COVID RBD and the nanobody. So you can perform this type of analysis also. And further, if you have the docked protein or model protein, and as you model the protein, so because you model the protein, you have to see the stability, further the stability of the protein. So you have to do the molecular dynamic simulation. So for this, there are different online tool also and the standalone tool also. So you can perform the molecular dynamic simulation of the model protein or docked protein or protein-protein interaction. And RMSD is just to see, like for structure model protein, ProCheck and other validation tool is just to check your protein model is accurate or not. So after simulation, RMSD is just to check your simulated protein is stable or not. Here you just check the plateau, like for this protein, here the x-axis represent the nanosecond in time in nanoseconds. So you have in this graph, just we check the plateau where our protein reached the plateau in the particular time frame. And this is just different types of analysis which you can perform after you model the protein. Okay, so yes, I'm done with the analysis and how you can validate your structure. So if you have further question, we can discuss, and that's all from my side.
Indeed, it was a great session, Dr. Poonam, and very quickly and very confidently you mentioned all of the validation of structure part. So, few are the questions in the chat box from participant related to our discussion topics. So, one by one, we will take this. So, the first question is from Safia that ma'am, if result is coming fail by verified 3D for any protein model, what should be our next approach? This is the most obvious, problem when we go for structure validation. Verified 3D or erect usually give maybe sometime that is fail. Structure validation is fail. So
Yeah.
this is very important question. What one should do after that?
Okay, this is the other tool which I explained.
Mhm.
So, as we model our protein from the Swiss model, okay.
Yeah.
So from there also we get the Ramachandran plot. This is just a confirmation. So I think we are getting the first step for any structure validation is the Ramachandran plot, because here we are checking our phi psi angle, all the residue are coming in the allowed region of Ramachandran plot or not. So forget about other types of analysis. Yes, we have to perform, but first what after modeling what we have to check is the Ramachandran plot. So I think you are getting the Ramachandran plot from the structure assessment of the Swiss model server also. So this is the just a cross check of the Ramachandran plot, and I don't have that Ramachandran plot saved result now, but the result was saved. So yes, you have to do other analysis, but you can validate your model structure by the Ramachandran plot. You will be very sure if you can look to the Ramachandran plot, and if all the residues are coming on the allowed region and favored region, you can say by yourself like your structure is accurate. And to validate that Ramachandran plot accuracy, we do other type of validation step. So one server will not work, but I think there are lots of server included in the web server. You can do all the other types of analysis for this, but Ramachandran is the basic for the analysis, and without any explanation, by looking on the graph only, you can check your model structure is accurate or not.
Okay, another question from Dr. Aditi Nag that warning and error message meaning in project analysis. I mean, she want to know how important this error and warning message in project analysis.
Where? Okay. Okay, this.
Yes.
Uh,
Hmm.
I think this is just a warning. You can ignore. The result is this. So that is not important. That is just a warning. In bioinformatics, you will get warning and error everywhere. But you have to, if you are doing the pro check for the Ramachandran plot, yes, because we are using the server, it's also using something. So it is just showing the warning because we are submitting our model structure. So you just have to see if it's more warning, that is okay. You have to see the result. You are using the pro check for the Ramachandran plot, so you just go for the figure. You can ignore that warning.
Okay, so one of the last question from Krunal that, ma'am, can we get this PPT? So Krunal, this question I will take. I will ask ma'am, Dr. Poonam for the PPT, and if there is no any copyright issue, then I will sure send it to you. Not to you, rather all the participants. So I will speak to Dr. Poonam maybe personally, and then we'll do that, right? So if anyone have any other question and query, so you may ask just for 5 minutes. So, and if not, then okay. So Abhishek Chahar is asking that oh, so it's again about the PPT. So yes, Abhishek, we will try to manage this if possible, right?
Okay.
So I
I
think
think that
no more question.
Sir, I think that's all.
Yes, yes. So I would like to convey my sincere thanks to Dr. Poonam for taking such a wonderful session, rather two sessions, and these two hands-on training sessions, we can say one of the core of this workshop. And thank you so much, Dr. Poonam, once again for accepting our invitation and giving your time and effort to this workshop. And I sure on behalf of organizing team that we will keep in touch with you for further your expert suggestions and something. And hand it to Dr. Mansita if you want to add something.
thank you so much.
Yeah, thank you.
Poonam ma'am, thank you so much. Our indebtedness to you for taking two of the sessions. That too, you are in a very, very different time zone. In spite of that, ma'am, thank you for managing for us. Thank you so much. And we'll definitely continue with our association in future also, ma'am. Thank you so much. I'm really indebted to you. Uh,
Thank you, ma'am.
yes, ma'am. Thank you. Thank you, ma'am.
Okay. Thank you.
Okay, so bye,
Uh,
Dr. Poonam. You may leave now.
Bye. Yeah.
I would request all the participants to please stay back for a moment. Participants, you all would have received an email mentioning the link for the valedictory session. So I request all of you to please leave this session and join the valedictory session. I request any of our organizing team members to please be in that session so that I'll be here till the end. See to it that all the participants have left this group. Any of our members of the organizing team, please join the other session, the valedictory session. Ravi Ranjan sir, Manali ma'am, Ravneet ma'am, please leave this session. We can join the valedictory, sir.
Sure, ma'am. So
Okay.
we are leaving this session and
Yes.
As you suggest, ma'am, we'll do accordingly.
Okay. Arvind sir, please close this session and start with the valedictory session, sir.
Yes, ma'am. We are just starting.
Okay, sir. Thank you.